All right, good morning, everyone. It's wonderful to see you on what hopefully is a beautiful and maybe even early Friday. Welcome back to day 2 of the Remote Neuropsychological Assessments and Research, Present and Future Needs workshop that is put on by a number of the institutes at the National Institutes of Health. If we could have the next slide, please. My name is Adam Hartman. I'm one of the program directors in the Division of Clinical Research at NINDS, the Neurology Institute. And I think we all met yesterday, so we're thrilled that you're here. And if we could have the first slide. We're going to just sort of go over some basic housekeeping stuff again.
Oh, 1 moment.
Do you guys need me to share those slides?
I have it 1 moment.
Okay. Great. So I'll just describe myself real quickly. I am a middle-aged white male with dark hair, glasses, beard, and mustache. I'm wearing a blue shirt and a blue and green regimental tie. So it's great to see everyone here. The basic housekeeping stuff, please remain muted unless you're directly called upon. That's going to happen only in the breakouts. Questions and comments should be placed in the chat. We will be engaged in some number of discussion periods through the day, so please do use the chat for that. And as I've said, the interactive discussions will be in the breakout rooms. And then if you have technical difficulties, you can reach Derek Smith, who is labeled as the RLA Tech, or email him at derek.smith@roselyassociates.com. I think with that, yesterday was really a fantastic discussion of some of the issues that we had regarding validation, regarding different populations, challenging populations, regarding how we can start thinking about looking forward in terms of equity and in terms of decentralized clinical studies. And so we're going to be sort of moving a little bit more in that direction this morning. But what we're going to be starting off with is a really exciting data blast for abstracts were accepted. Each person is going to have a few minutes. Each presenter is going to have a few minutes to present their material, and then we'll have 1 minute of clarifying questions if there are any after each presentation. And then we're going to have a few minutes, hopefully, left over for more of a group set of questions, group discussions, maybe some cross-cutting themes. So with that, why don't we go ahead and start? And I'm just looking to see if we first is going to be Bridget Kelleher. I'm going to ask each of the presenters to introduce herself. So our first one is Bridget Kelleher.
All right, just get started and dive in then.
Jump in, the water's
okay.
warm.
Great, let me start my timer. 3 minutes will go by fast. Hi everyone, I'm Bridget Kelleher. I'm an associate professor of clinical psychology and neuroscience at Purdue University. For those not watching visually, I'm a young to middle-aged white woman with dark brown hair. I'm wearing a pink jacket and a black shirt. So I'm going to share with you all today a study called PandaBox, or Parent Administered Neurodevelopmental Assessment, and how we're using this protocol to remotely monitor a variety of high-risk youth. We'll progress. There we go. So PandaBox was developed prior to the pandemic through a K-23 award as a method for standardizing remote assessment procedures for collecting integrated biobehavioral data without an examiner on site. And so what I mean by that is we shipped this kit to families across the country. Our primary projects are focused on families who have children with rare genetic syndromes like Angelman or Fragile X syndrome. And we remotely coach the families through a variety of tasks that are typically done in lab. What makes PandaBox a little bit unique is that we're not just doing behavioral tasks, but we're also collecting audio data that's automatically kind of counting some different variables via the Lena device, which was mentioned yesterday. We also collect heart activity data from both the parent and the child, and we're integrating all of these data so that we can look at some really interesting things. These data were published in 2020, and I have a QR code if folks want links to the papers that'll be up on the screen the whole time. So here are the tasks that we do for PandaBox. We look at attention and eye movements. This is a task where we look at both behavioral engagement with a video and also how a child's heart rate is changing as they watch a video task. Heart rate decelerations have been used for decades to index cognitive engagement in infants, and this is something that we can do 100% remotely. We also look at temperament, developmental play, social communication, and parent-child interactions, and we're coding a variety of different social communication and attentional and developmental variables across those tasks. We've collected over 200 PandaBoxes, and we're wrapping up data collection in December for the primary project. We've done this in children with genetic syndromes, infant siblings of children with autism, and we've even tried some of these tasks with infants with congenital Zika syndrome in Brazil. We're finding that families really like this protocol. They do a really nice job doing the assessments. What's nice is they have high fidelity because we're live coaching them in real time. So if they do something wrong or have trouble, we can coach them on redoing the task. The infants are engaged and the caregivers have been pretty satisfied across a number of different syndrome groups. We're also expanding this now. So we have an EEG expansion where we're looking at power spectra that we're collecting entirely remotely in children with Angelman syndrome and their families. This is work being led by Dan Fody here at Purdue. And we've been able to show that we can capture the increased delta power that's common in Angelman syndrome. Again, doing this entirely remote setup. 88% of caregivers prefer this over their local or clinic options. And we like to share. So we're very committed to open science, and we received an R21 to develop a hub to help train other sites on PandaBox. This is getting ready to launch, and also a new software tool that can be used to process those heart rate decelerations that I mentioned. So if these are tools that would be helpful for your team, please contact us on the QR code. There's a link where you can, we'll email you some resources, and we're looking for beta testers. And that's all. I'll take any questions.
Fantastic. Thank you, Bridget. Any questions in the chat? I have one quick question. What are you using the EEG for? Are you using that EEG during the task or during a specific set of tasks, or because there's a fair amount of data out there already?
Yeah, so this is just designed to capture kind of resting state in that the children have a kind of boring video that's on the screen during the task. We're not at this time trying to do ERPs, but we do have a grant to look at whether that's possible. Our guiding feature is that we try not to push the constructs past the tech. And so we want to make sure that the tech is precise enough to get at those more temporally precise variables, but it's doing a nice job. The setup is working well for looking at basic power.
Fantastic. One other question, are you testing a normal population or comparing to a gold standard for validity?
That's a great question and a complicated one because we're doing a lot of different things. We do have a typically developing control group that does PandaBox 2 that we have longitudinal data on, so we're able to see if there are any kind of differences. We talked a lot about gold standards yesterday. I think for a lot of the things we're doing, especially in the rare disease space and the rare disorder space, there aren't good gold standards. So we are looking at concurrence with a variety of other parent report and other types of tasks that we can collect remotely, but we're not comparing this to something like the Bailey, in part because it's not designed to really replicate the Bailey. But we did beta tests where we had families do both remote and in person just to make sure that the procedures seem to align. But kind of validating against a developmental assessment isn't a primary priority. We're interested in developing kind of new metrics that can be used to measure cognition over time.
Fantastic. Right. Great. Thanks a million, Bridget. We appreciate it. Thanks for your talk yesterday, and thanks for the data blast. Our next speaker is Nikki Stricker. Dr. Nikki Stricker. Nikki.
Thank you. Let me just start sharing here and then do a swap. Let's see. All right, are you seeing my correct screen?
Yes.
Great, thank you. So I'm Nikki Stricker. I'm an associate professor of psychology at Mayo Clinic in Rochester, Minnesota. My funding disclosures, or my funding sources and disclosures are listed below. For those of you listening in without being able to see me, I'm 5'1", white, middle-aged with light brown hair. I'm wearing a black suit jacket and glasses, and my pronouns are she, her, and her. In the Alzheimer's field, there's a clear need for efficient methods to identify individuals with MCI and dementia who may be a candidate for treatment. Remote, highly scalable, self-administered cognitive assessments are likely to be a really important aspect of early identification. Mayo Test Drive is a web-based cognitive testing platform that is compatible with smartphones, tablets, and personal computers that we developed in 2020. And we specifically designed it for unsupervised remote self-administration in both clinical and research settings. The screening battery takes about 15 minutes to complete and includes a measure of verbal memory and a measure of processing speed and executive functioning that together form the Mayo Test Drive composite. In the lower right corner of the slide, I provide a brief summary and QR codes of our current publications that support initial validity and psychometric properties of Mayo Test Drive measures. The objectives of the current study were to investigate continuous associations between remotely administered cognitive measures and amyloid and tau PET biomarkers, which are considered core biomarkers of Alzheimer's disease. We also wanted to determine if remote cognitive measures show lower performance in MCI and dementia participants compared to cognitively unimpaired individuals. This study included 684 older adults who were predominantly from the Mayo Clinic study of aging. Most were cognitively unimpaired. 34 were diagnosed with mild cognitive impairment and 7 with dementia. We had an older adult sample. The average age was 70, and participants were primarily non-Hispanic white. All but 3 individuals completed Mayo Test Drive remotely and unsupervised. And as you can see in both the regression results in the table at the bottom, as well as the scatter plots, lower performances on the Mayo Test Drive composite were associated with higher amyloid tau and interinal cortex tau. The Mayo Test Drive composite also did show a similar pattern of associations as the in-person Mayo PAC. There are subtly different sample sizes across these remote and in-person measures, so we didn't want to directly compare them, but I wanted to show these values for reference. The graphs displayed are scatter plots, and these illustrate the associations between the Mayo Test Drive composite and each of the 3 imaging biomarkers. The text is a little bit hard to see in the figures, but the color-coded boxes correspond to the table labels below. The point of the figure is really to show that associations are stronger when limited to the MCI and dementia participants only, and that you can see on the right. Associations are still significant, but are weaker in the cognitively unimpaired only participants on the left. As one example, for amyloid PET in the top row, the correlation coefficient is -0.24 for cognitively unimpaired individuals and -0.52 for those with MCI and dementia. I'll briefly summarize the last slide and then also the last set of results here. So remotely administered Mayo Test Drive measures show significant associations with core AD biomarkers. These associations are more robust in participants with cognitive impairment, but they remain significant in cognitively unimpaired individuals. As you can see in the data presented on this slide, MCI and dementia participants show lower performance on the Mayo Test Drive composite and subtests compared to cognitively unimpaired participants, and this effect size is large. The same pattern is also observed when we limit to mild cognitive impairment participants only, and when we adjust for age, sex, and education. Overall, these results provide further support for Mayo Test Drive criterion validity and provide evidence for likely clinical utility. Please contact me with any questions, and I'll provide links to the published papers mentioned in the first slide in the chat for those who are interested in more details. Thanks very much.
Fantastic, thank you. We had 2 comments, and I think we might want to take them up during the cross-cutting discussion period. One question I had, Nikki, was you showed a phone, and maybe I missed this. Are people using their own phones? You send them the phone? How does that work?
Great question. This is a very, in the wild, it's both a bring your own device and choose your preferred device, because you can also do it on tablet and personal computer.
And have you looked at differences between different platforms, or?
Yeah, we've developed normative data, and as part of that, we included device as another variable to be included in the model. To be included in the normative model, we set a standard to explain more than 1% of the variance, and device didn't reach that standard. So I will always say there's going to be device differences. I was reassured by our data to show that it wasn't explaining more than 1% of the variance.
Fantastic. Any other questions? I don't see any in the chat. Like I said, there's some issues about validation that I want to bring up with everybody. Fantastic. So our next presenter is Dr. Vicki Levitt.
Thank you. I will share my screen. I want to thank Adam and the organizers for inviting me, and I find it a bit daunting, actually, given the 2 quick data blasts we just got. They were so impressive. This is not so much a data blast as a prototype blast. It's very early days. Before I begin, I will say that I am a white middle-aged woman with long brown hair, and I'm wearing a blue blouse. And so I'm going to talk about a tool that we designed in my lab called the Language and Memory Test. It's an app-based composite cognitive measure. We designed it to address a very specific problem in multiple sclerosis, but I do hope to be able to use it in other populations as well. I am an assistant professor of neuropsychology in the Cognitive Neuroscience Division at Columbia University in New York. So the problem that we're trying to address is detecting cognitive change over time in MS. MS is a population that has a very early onset in early adulthood at an average age 32. So folks who are very high functioning individuals come in and they say that they're experiencing cognitive changes, but our gold standard clinical tools are not very good at detecting this. And really what we've been using are tools that were designed for other clinical populations and just using them in MS. So there's a need, and the problem is quite underscored by several publications that have come out, which I've included on this busy slide, but all of them are making the same point, which is that we have biologically implausible changes in cognition over time. That is, cognition is improving. So we're seeing that in this first study here, the PACEAT is showing steady improvement in concert with increasing disability and decreasing mobility. The same thing here with the SDMT and the PACEAT in large clinical trial data. And that's the same thing is seen for placebo as the treatment groups. Yet again here, and then in a paper that we have in preparation, we're also seeing that these improvements in cognition across cognitive tests are accompanied by increasing atrophy. So what we wanted to do was leverage technology to create a superior tool. And to do this, we need to overcome the practice effects and regression to the mean, which are probably responsible for what we saw in the prior slide. First, we need to identify the cognitive domains that are most sensitive to the changes in MS. And we need to then leverage technology to minimize practice effects, as well as develop a tool that's well tolerated by everyone who's going to use it. So I want to walk you through my prototype right now, which really just seems a little bit rudimentary compared to all the beautiful data we just saw in the 2 prior talks, but I'm going to bring you along for the ride. This is what we created, and this is what we're rolling out right now. We call it the LMT, which stands for Language and Memory Test, the 2 areas that my data from my lab show to be the most sensitive to change over time, specifically lexical access and visual spatial memory. So the test is about 3 to 5 minutes long. It includes 3 different subtests. The first is finger tapping. So you have 10 seconds to tap that button in the middle of the screen as fast as you can. And then the next task has you seeing an item on the screen that you must say out loud. The microphone picks up your response and advances to the next screen, which shows you that image in a grid. You're only shown that for a second. Notice that every detail is manipulable, so we can increase that stimulus presentation time or decrease it. You're walked through 9 different stimuli, and then you're tested. Now you must show us where you saw that item. At the very end of this, there are 3 subscores that we're provided with. The first is how many taps, finger taps. The second is the time it took you to produce that verbal response. And the third is your accuracy on the memory test. So we're beta testing this now. We're going to be collecting normative data. This conference over the last day has really driven home the need for validation, proper and rigorous validation studies, which are all planned for the future. In the meantime, we have a very exciting opportunity to incorporate the LMT into an ongoing clinical trial of a micronutrient fortified cereal being given to adolescent girls. This is a trial being conducted jointly by the University of Ghent and Arba Minch University in Ethiopia. And these researchers are very excited to incorporate the LMT into their study. So one thing I might not mind talking about if we have an opportunity is how early days is too early days to go ahead and beta test and validate a tool in the field. And I think I will leave it right there. I don't have an acknowledgement slide, unfortunately, because my desire to be a rule follower is too strong. And so I didn't include one. I was only allowed 3 slides, and that's what I have. But I have many people to thank in the future. Thanks, Adam.
Victoria, take a minute to do your acknowledgements, at least verbally.
Okay. I want to acknowledge a young man named Sean Trainer, who is an undergraduate at Tufts University, who built this app for me over his Christmas break last year on a shoestring budget. Many people will tell you that apps are extremely expensive, and they can be, and they can also not be. So Sean is the first person. The other person I want to acknowledge is Alex Ratzner, who was a data tech in my lab, who introduced me to Sean and supervised Sean. He's now a PhD student at NYU in the computational neuroscience program. Those are the two people who made this project possible. There was no funding.
Fantastic. So 2 quick questions. One is, do you tell the patients what you are measuring?
So you just saw exactly what the patients see.
Okay. Great. And then how did you determine which domains were most sensitive to MS?
I looked at my longitudinal data from several hundred people, and I also looked into the literature. And my read of everything is that these, and by the way, the gold standard for me is what patients say. So, I see patients 1 day a week, and they constantly talk about their difficulty getting the words out. And oftentimes they say, I have memory problems. And my follow-up question, give me an example. Well, halfway through a sentence, I can't remember what I was trying to say. Actually, I can, and I know what I want to say. I just can't get the words out. So that brought us to focus and really hone in and actually develop a paper and pencil test of this rapid naming efficiency that has proven to be very sensitive to when people describe this issue. And it also correlates with cortical thinning in language areas. So this is what drove me to create this test.
Do they know they're being timed?
Other than the direction to name this object as quickly as you can, they don't. Now, I mean, you're asking me these questions. Do you think they should? Those are things we can manipulate, right?
These are questions from the chat. So.
Oh, interesting. Okay, yeah.
Yeah.
They're my questions too. And the awesome thing about digital technology is we can manipulate every single one of these things. We can give people more or less instructions. We can give them more or less help along the way, more or less feedback as to how they're doing. There's so many different elements we can unpack here.
Great. So you just answered the other question, I think. Fantastic. Well, thanks a million. We want to leave time for Dr. Jessica Kennard. Jessica, you ready?
Yes, ready.
These have been fantastic. I'm getting private comments. These are all fantastic.
I'm going
Go
to
for it.
share my screen.
Please.
So I'm a white female brunette wearing a necklace. Let's talk about experiences of Spanish-speaking families with a remote neurodevelopmental assessment. We just heard from Dr. Kelleher about the parent-administered neurodevelopmental assessment, or Panda Box. For our study, we culturally adapted and translated the Panda Box into Spanish and interviewed families about their experiences. Okay, so first, why is it important to complete a formal translation and cultural adaptation of a remote neurodevelopmental assessment? Couldn't an interpreter or bilingual clinician simply translate as they go, which is what is typically done with remote assessments for Spanish speakers? Well, there are several problems with that approach. During the pandemic, it was found that Spanish speakers are significantly less likely to use telehealth than those with English proficiency. Relying on in-the-moment translation introduces confusion around difficult to translate topics, not to mention the additional challenges of trying to coach parents through an interpreter over telehealth, with some clinicians reporting that they dread this type of telehealth interaction. Beyond language issues, telehealth creates barriers for families with limited resources, such as limited experience with technology. These barriers are particularly unfortunate when thinking about Hispanic families, because telehealth could actually provide a way to reduce disparities for Spanish speakers. For example, in my field of autism research, children from Spanish-speaking families get diagnosed with autism significantly later than English-speaking children due to barriers such as lack of bilingual providers and difficulty traveling. What if families could get access to Spanish-speaking providers without needing an interpreter and without needing to travel? As another example, Spanish speakers are routinely excluded from research. What if we could send the developmental research lab to families? So for our study, we had 2 aims: to culturally adapt and translate the Panda Box into Spanish, which we vetted with 10 native Spanish speakers, and to explore the feasibility and acceptability of the Panda Box among 13 Spanish-speaking families with toddlers at elevated likelihood for autism. Here's what we found out. In terms of feasibility, all mothers value directly speaking to a specialist in Spanish. For most families, technology was easy to use with the extra support we built in during the adaptation process. When a tripod was added, parents felt the video adequately captured their child's behaviors. We culturally adapted peekaboo and book reading activities to better match families' cultural experiences. In terms of acceptability, parents liked administering the assessment, with many feeling that the natural environment provided a better view of their child's behaviors than at the clinic. Many preferred the Panda Box to in-person. And because we so often use questionnaires in our work as researchers, I also want to point out that many of our families had difficulty reading, even in Spanish. So for these families, playing with their child in the Panda Box was viewed as easier than filling out questionnaires. In summary, the Panda Box Spanish provides a promising option for increasing access to remote neurodevelopmental assessments for Spanish-speaking families. If you're interested in brainstorming ways to get Spanish speakers involved in your remote work, you may email me or scan this QR code. And I also want to acknowledge Dr. Kelleher for developing the Panda Box and Casey Okonowski at RTI International for helping with this work. Thank you.
Fantastic. So I had one quick question, and that is, and then we'll do a group discussion for a few minutes. One quick question. So so Spanish is not Spanish is not Spanish. And so do you have, there are dialect variations between different communities. And so how do you take that into account when you're thinking about that?
When we translated this, we went through a really rigorous process where we had 2 separate groups translate. Then we synthesized the 2 versions. Then with that synthesized versions, we interviewed 10 native Spanish speakers who were from all sorts of different countries.
Okay.
Get their feedback on the language. And then as we were trying it out with the families, we asked them about the dialect. And actually, even after all of that translation, we still had families saying, don't use this word. I would never say that to my child. It's offensive. So it was a really important process. And in the end, families were saying that it was understandable and clear.
That's fantastic. Good. Could we do a quick panel setup? I'm actually asking the RLA folks if we can do that with our Data Blast presenters. These were fantastic. Like I said, all 4 of them, I'm getting all sorts of private comments about how wonderful this is. And so thank you all for presenting your data. We have a couple of minutes. And so one cross-cutting theme, and this, I was thinking about this, and then Luke Stokel actually asked this question. So for group discussion, validation is clearly important. Would be interested to hear more from the group about fit for purpose use and validation for remote assessment, how it's reconciled with the different tests, batteries, devices, platforms, apps, et cetera, used across all the different purposes for use, how best to harmonize across studies if we can't control test and battery versions, devices, platform apps, et cetera. Are there novel methodological approaches we should be considering here for cross-study comparisons for within-person comparisons? Who wants to take a whack at that first? All right, Victoria, it's up to you. Take us out.
I want to ask Luke if he wants to collaborate with me. So here's what I think. I think those are all such great considerations, and it makes me want to lie down and take a nap because I am not an expert in psychometrics. I had a vision and a desire to build a tool for which there's a need, but I don't know how to do all these things. So I think we need to have mechanisms for collaborative, collaborations that can allow people with different skill sets to come together. I think that's why I'm here. And that's what I'm hoping to get from this is get it connected to the people who can do those things that absolutely have to happen. That's my quick thought.
Fantastic. Who'd like to go next?
I'll just quickly add for our study, we collected validation data alongside the qualitative interview data, which we're still analyzing. Because we did this during the pandemic, it wasn't in person. So we were comparing it to other remote questionnaires and so forth that have already been developed in Spanish to compare what families we're reporting on the questionnaires and what we measure in the Panda Box. So that's forthcoming.
Great. Next, Bridget. Yep.
Yeah, this is such an important question, and validation and the psychometric interrogation of these tools is super critical. I do think there's a step that's often missing when we go to remote assessment, and that's the user experience step. So that's what we were really trying to tackle with PandaBox is not only do the measures align or correlate or predict or whatever that, those questions are, which are important, and we're doing those now. But what we presented today and what took years was just to figure out how do we even do these things in a way that families find them acceptable, that patient communities can engage with them. And I think a lot of times when people try to flip something remote, they skip straight to psychometrics. And so while those things are critical, I think a lot of the projects that are flipping over are still in that early stage of user experience. And we don't even really have like a nomenclature for that. So I don't mean to devalue validation. It's critical. It's the next step. And I think a lot of projects are still at that pre-step of how do you do this in a way that your entire participant has changed when you go remote. It's often for us, it's the caregiver now instead of the child in terms of who's engaging with us. So it's really a different method.
Nikki, you want to be the last word on this, or next
Sure,
to last word?
I can just comment that this is well, there was a lot there. The part that I can highlight is it's both easy and hard to share digital measures. So it's easy in that if you use the platform it was developed in, then it can be easily shared once you get through the different technology sharing aspects. But then it's hard if you want people to only have to go to one system. So then if you want to share, you need to make your code open source or share it with that group. You have to make sure they built it again and exactly that you did, etc. Jason Hassenstab, I'll mention, who's a great remote assessment researcher. We took his test, the symbols test, because he provided it open source. So that's one of our measures, and then we developed one of them. So that was a good example of a tool being made available. We implemented, he checked that we did it right. And so there is definitely the capacity to do that. There are nuances along the way, but it can be done.
Fantastic. So it sounds like we need to wrap up. It sounds like the psychometricians, if they want to collaborate with anyone, have plenty of people to collaborate with. And that's, quite frankly, part of what we hope comes out of this workshop is that if you see someone presenting something, by all means, get in touch with them, because that's, collaboration's the next big step. So with that, thank you very much. We're going to move on now to the next part of this morning's session, and that's welcoming remarks from two of our partner institutes at NIH. And so we're going to be teeing up some welcoming comments, some video comments initially by Dr. Josh Gordon, who's the director of the National Institute of Mental Health. He oversees an extensive research portfolio of basic and clinical research that seeks to transform the understanding and treatment of mental illnesses, paving the way for prevention, recovery, and cure. So with that, some comments from Dr. Gordon.
Hello. And thank you for giving me this opportunity to provide some remarks on the outset of the NIH Remote Neuropsychological Assessments and Research Workshop. I'm Joshua Gordon. I'm the director of the National Institute of Mental Health. I'm sorry I can't be with you there today in person due to another obligation, but I wanted to stress to you why we think this workshop is so important for the work that we do at NIMH and really for all of NIH. First, why are we interested in neuropsychological assessments in the first place? Well, we recognize that mental disorders are complex and vary from one person to the next, and that our diagnostic system are incomplete descriptions of our patients, number one, and number two, at best only partial predictors of important clinical phenomenon such as response to treatment and future course of illness. We believe that a better description of the specific cognitive and behavioral abilities and disabilities in our patients will inform our understanding of mental illness, which parts of the brain and which aspects of brain function are disrupted in those illnesses. And importantly, they will help us further describe our patients in ways that are clinically relevant. So neuropsychological tests, measures of cognitive and behavioral function, they're designed to tell researchers and clinicians about how the brain works. They capture processes like motor abilities, specific cognitive functions, specific, behavioral aspects that are impaired in many mental disorders and that are probably closer to the natural substrates of brain function than the diagnosis themselves. So why are we interested in remote testing? Remote measures can be accessible and scalable. What do we mean by accessible? We mean they can reach people who otherwise would have challenges participating in clinical research, be it people in low resource urban or rural areas, people with disabilities who have other barriers to travel, people who live far from the typical laboratory settings where in-person testing can be conducted. Of course, if we truly want these tests to be accessible, it's important that we address challenges with implementing remote measures in underrepresented cultures, in languages that aren't English, and in individuals who have challenges in terms of accessing high bandwidth or high capability devices. So we need a baseline of remote testing that can reach as many people as possible. I said that these remote tests are accessible. They're also scalable, meaning that it costs relatively little to administer these tests on a large scale, and that there's a lower burden for participants and their families. This efficiency, this lower cost, allows us two major advantages. Number one, we can get these data on really large numbers of people. I'm talking thousands, hundreds of thousands of individuals, and use that data to better refine our understanding of a wide variety of patients from a wide variety of backgrounds. Second, this low burden, low cost allows for the capacity for longitudinal measurements. Over and over and over, we hear from our scientists that we need to understand better the longitudinal course of illness, the longitudinal effects, of treatments. We need to understand things in the context of an individual and how they change over time. And these low burden, low cost tests can be administered over time longitudinally to lots of people. There are lots of other reasons why we're interested in these remote tests. The potential for reducing bias by multiple measurements, the remote assessments that can be more easily perhaps tailored for specific populations and contexts, the opportunity for decentralized clinical trials, lots of other wonderful things. But bottom line is we are really interested in developing and a library of remote tests that measure cognitive and behavioral function that we can administer at scale and that are overall accessible. What are we doing already with these kinds of tests? Well, in combination with the All of Us research program, which, as you know, is a cohort of hopefully 1 million people, 1 million Americans, a diverse cohort, that all of these individuals agreed to donate health information, electronic health records. They answer surveys, they get their genome sequenced, and we are rolling out cognitive and perceptual tasks through the Exploring the Mind program to as many of these individuals as possible, so we can map performance on these tests onto the existing clinical data that already have, onto the survey data that measure mental health function in a variety of ways. Pilot testing began in this program in December 22nd, 2022, and as of mid-October 2023, with the full launch in September, there are more than 20,000 completions for each task, and the numbers are rising. To supplement the All of Us research program, we've also launched the IMPACT program, or Individually Measured Phenotypes, to advance computational translation in mental health, IMPACT MH, as part of our overall initiative in precision psychiatry. This program will replicate what All of Us is doing in their general sample in cohorts of individuals where people can bring together these behavioral measures, clinical records, and other information about our patients to be able to understand how much better can we do at clinical prediction in clinical populations than we can do with these tasks than we can do with diagnosis alone. Finally, we're encouraging the use of remote assessments in clinical settings. including things like the Penn computerized neurocognitive battery, which has been adapted for large-scale data collection across the United States, the Early Psychosis Intervention Network, or EpiNet, which has similar batteries that are being used in individuals with first episode psychosis. A lot of different studies that use remote testing of neuropsychological functions, and we need more. We need more tests. We need better validation. We need better adaptation to different communities to increase the accessibility, to increase the reliability, to reach those that we want to reach, so that we can help those that we want to help, and that's people with mental illnesses, communities with mental illnesses. So I thank you for getting together at this workshop to talk about how to advance this field. I wish you lots of luck, and I can't wait to learn what you all uncover. Thanks. Bye-bye.
Well, thank you, Dr. Gordon. That was fantastic. I think we have met the challenge of producing some very interesting questions, which is, a lot of times the best responses to develop some really good questions. And so now it's a real pleasure for me to introduce another one of our sister institutes, National Institute, the Eunice Kennedy Shriver National Institute of Child Health and Human Development. We're really pleased that Dr. Allison Cernich, who is the Deputy Director, is with us today to give some live comments. She became the Deputy Director in September 2019, and in that role, she assists the Director of NICHD in overseeing the Institute's programs. Prior to that, Dr. Cernich was the Director of NICHD's National Center for Medical Rehabilitation Research, or NCMRR as we all know it. Dr. Cernich, welcome and thank you.
Thanks, Adam. And I have to say I don't queue up my NIH video cast queue very much for my Netflix viewing, but I will tell you that I am actually going to watch this whole webinar when I get a chance. I really want to thank NINDS for planning this important, timely workshop. By way of background, I am a board certified neuropsychologist, and my early career way back in the day was focused on remote neuropsychological assessments in clinical and research scaling. And I really resonated with what Dr. Gordon just said. One of the initial ways that we used remote neuropsychological assessment back in my early research was with the military. And these were scalable technologies that we could use across different service organizations, looking at large scale screening for what was then sort of the newly burgeoning epidemic of concussion. So these aren't new technologies, but I think COVID also brought into stark relief the fact that the need for these technologies to be remote, to be accessible, to be able to be scaled is really something that we need to address and have needed to address for many years. And I think the other thing that's really important, and it was brought to the forefront in a number of places over the past couple of years, is that many populations have really been underrepresented and understudied in research utilizing remote assessments. And this doesn't just include minoritized racial and ethnic groups. We also have these folks that have limited access to technologies. And also, as was just discussed, people whose primary language is one other than spoken standard English. And for NICHD, our priority populations are often really overlooked when it comes to developing these technologies. And when I say that, I mean children and persons with disabilities, including physical, intellectual, and sensory disabilities. Thinking about universal design with respect to these technologies may actually make them much more adaptable across different populations. And so I think, there's another a piece of this where we have to think about how accessible are these technologies, how adaptable are the methods that we're using. So if I'm a neuropsychologist and I'm trying to test somebody, if I'm in the room with them, I can adapt that test and I can try to make it more readily available to them if they have a hearing impairment, a visual impairment, a motor impairment. If I'm using a technology, how am I going to make that more accessible? And I think that poses a different engineering, different hardware, different software, different interaction challenge that I think would be a really fun mental exercise for us, but also a great scientific opportunity. In addition to the needs of the persons with disability, there's this added layer of complexity. if we are testing children who may be the primary English speaker in their family, we may have some translation issues when getting consent from their parents or working with their parents, and or conversely, where the child is going to be a translator for an older adult taking these tests. And they are really going to help to fill in the gaps for those adults that need assistance due to language issues or other assistance needs, which also comes up in validation, right? And identity validation in particular. So, the members of these groups really should be involved in the development of the remote tools and understanding that UI UX sort of question, right? The user experience and the user interface. How is that going to be factored into the platform? And also how does it get factored into the validation process? The other thing that I think has been noted is that when you adapt an in-person test to virtual delivery, it's a new test, right? And we've been saying this for years. It's just something that we know that the traditional gold standard test is not going to be the better one or the more valid one. And we even know that in our traditional neuropsychological practice, that the gold standard tests don't do well for certain populations and was a major source of controversy last year in the field. The change in format also introduces technical changes based on the platform. And this is something that we noted a very long time ago. Depending on how you are putting this into play, you really need to make sure that the performance of the test is going to be compatible across platform systems, the ways that you do this in perspective of the user experience, but also in the form of the validation of the test and its integrity. So, I think that this community, it's so great to see some of you here because, we can address some of this proactively. And really what we do if we don't do this, if we don't proactively address some of this, these tools will exacerbate known disparities and underrepresent marginalized populations in the future research. So, we really need to be intentional about our outreach and recruitment. We really need to think about how we address these disparities and how we monitor for these issues. And we really have some good news in that we think that some of these hardware and software tools are commercially available now, and we have the potential to overcome some of these barriers. So I just want to say thank you to the planning committee. This is such a research area that's so near and dear to my heart. It's something that I've worked on my whole career. And I really look forward to seeing what innovative things that this team can come up with and how we can help you at the leadership level partner across communities so that we can bring our engineering friends, our psychometrists, our psychologists, and the folks that have the subject matter expertise together to make a difference in this particular field. So thank you so much. I hope I've caught back a little of your time. And Adam, really appreciate the invitation to come and give some remarks.
Thank you so much, Dr. Cernich. It was great seeing you. And just to highlight, so we're actually running a little bit ahead of schedule, which is good. So I'm going to just take this opportunity really to thank our sister institutes. And so I know you've heard from Dr. Korshitz, Dr. Gordon, Dr. Cernich. We also wanted to thank our other partners at the National Heart, Lung, and Blood Institute and the National Institute on Aging and the NIDCD, National Institute of Deafness and Communication Disorders, because they've all been a very, very important part of the planning process, the planning committee. They've had some very, very significant programmatic input. And so we wanted to thank everyone. This really is truly a trans NIH effort. And it's been a lot of fun planning it. And I have to say, it's been an awful lot of fun listening to all of the discussions. I don't know, for those of you who are following along in the chat, we've already had a request for a follow-up workshop. We'll see. This is the sort of thing that you don't do more than about 3 or 4 times a year. So it's takes a lot of effort, but it certainly is, I think in this case, worth it. And we'll talk about what the next steps are going to be. I don't know. We need to have a little bit of a post-mortem analysis too. So with that, what we're going to do, I think, is set things up for, we've actually bought ourselves a little bit of extra time now for the breakout sessions from yesterday, hearing the report outs from those breakout sessions. And so the format for this is that we're going to have each of the groups who were in the breakouts yesterday present the results of their discussion. Each of them is going to take about 5 minutes. And then we're going to have a cross-cutting discussion. And I think that some of the issues that came up in the chat earlier can also be further fleshed out a little bit, some of the issues that maybe were residual from yesterday, because it's just been, one heavy hitting talk after another. So just to briefly remind everyone of what the 4 breakouts were yesterday, I'm just gonna basically go through the topics. And while I'm going through the topics, for those of you who are going to be presenting the summaries, please start to tee up. So the way this is going to work is each leader, non-NIH leader, is going to be sharing their screen, and they're going to present their slide, and they're each, like I said, each going to have 5 minutes. So the 4 breakouts that we had yesterday are room 1 was validation of tools into different languages, and so that's going to be one topic. The next one is from my room, ensuring methodological fidelity and integrity with multi-site trials. The next one is safety and reporting issues, and the last one is equivalence versus validity and reliability, comparison between remote and in-person. And so this should be a lot of fun. I've already seen the slides, and I can tell you these conversations were fantastic in all 4 groups. And I think we're going to have, once again, we're probably going to have too much to talk about rather than not enough to talk about. We're really, it's going to be a fantastic discussion. And so the first person who I'd like to introduce, it looks like she's ready to go, is Dr. Liz Pena. Dr. Pena, you ready to share your slides and talk about your group?
Sure.
Go for it.
Click on share slide. Okay, there we go.
Yep, we're good.
Okay. There we go. So yeah, it was a great discussion, and I can say that like this whole theme of the workshop, there's a lot more to talk about that we had time to talk about. For basic questions that we were to our charge was to talk about gaps and needs with regard to remote assessment tools for speakers of other languages. We talked about existing tools, what were key issues in consideration for adapting and translating existing measures, and the key issues that needed to be considered for using remote measures of cognition. So just to kind of summarize what are existing remote measures that are appropriate with speakers of other languages, really the theme here is that there are very few measures. These are a few of measures that were mentioned, and I added one more this morning, but most are locally adapted. Most are developed for the purpose of a particular study and developed in the local context. Some of the key issues that we need to think about when we're adapting or translating existing measures is that not everything can be translated. Maybe not everything should be translated. We know that languages are structured differently. Frequencies affect how people perform on frequency of occurrence in a language, affect how people perform on a given test. And what we're doing when we're testing, we're using language, but we often are centering linguistic based measures to do the kinds of testing that we do. And so we really need to think carefully about what it is we're doing, what it is we're focusing on. Sometimes there's a tendency to center English and what's important in English, and that brings along a specific kind of cultural perspective. And so there may be threats to validity when we start with English and then translate to other languages, assuming that it's going to be the same. So we have to take into consideration different cultural perspectives, as well as the structure of the languages that we're trying to translate to. There are significant costs in terms of labor, time, methods in translation, and it is very difficult. I think earlier today there was this mention of, can we translate in the moment? Well, no, it's very hard to do that and to get valid and reliable data for our methods. Technology, however, allows for global access, and that's certainly a really important aspect of being able to do testing remotely. It allows for different persons to have access to these materials, to have access to the testing that we have available, to have access to research studies. And so that's really critical, but we have to weigh that with with careful translation, for example, and careful adaptation. Some key issues that came up in terms of considering remote measures were variations in technological literacy and the topic of perhaps if we're working with diverse populations, we may need to be oversampled. This should not be an afterthought or a checkbox on the NIH recruitment strategy, but should have careful consideration of when we're doing research with populations from diverse linguistic and cultural backgrounds, that we might consider oversampling to really figure out what it is that's going on. So we had three major, so those were our kind of our quick answers to questions. And our kind of major themes that emerged is to build for the specific purpose. We need to use ground up approaches, and it is important to leverage technology to be able to maximize the use of that technology. And I think we need to tailor to remote, to what remote access can buy us and what the technology can buy us. Beyond translation, we need to think about, which is usually focused on linguistic equivalence, we need to think about functional equivalence. So I mentioned that word frequencies, for example, across languages are different. What those represent functionally are going to be very different across languages and cultures. Markers, for example, in my area, I'm a speech language pathologist. Markers of language impairment are different across languages because different things in those different languages are early acquired, other things are later acquired, some things are more difficult in one language than in another language. And so we need to think about what that means for linguistically based measures when we're translating. So translation doesn't mean we're going to end up with functional equivalence, for example. How does this translate culturally? Is it going to be equally meaningful to the different populations that we're working with in the same way? Are they going to interpret the intent of the question similarly? And then finally, we need to think about psychometrically, how is this equivalent? There was a comment in the room that I think resonated with a lot of us, that technology can amplify human efforts. And so we need to think about intersection with the capacity that we have, think about universal design principles, and apply those to working with diverse populations, diverse linguistic populations as well. So that's kind of the summary of our discussion, and I look forward to further questions and discussion. Thank you. Now, if I can unshare.
That was while you're doing that, while we're transitioning over to group two, that was fantastic. And I think one of the things that I really love about this workshop has been that we've really managed to move beyond that sort of first very concrete layer of things. And we really are hitting on some all the groups are hitting on some very sophisticated and I think nuanced points that are really important that, you really need to think about moving forward. So thanks a million to group one. You guys hit the nail right on the head. So thanks a million for that. Our next presenter is going to be from our group, Dr. Svetlana Shore, and she's going to talk about, our group discussed ensuring methodological fidelity and integrity with multi-site trials. Take it away.
Hello, everyone. I'm Svetlana Shore. I'm a middle-aged white female with dark brown hair pulled back in a bun, and I'm wearing a purple blouse. In breakout 2, our goal was to discuss how investigators can establish and monitor methodological fidelity and integrity when using these remotely administered cognitive tools, specifically in multi-site trials. And we talked about what potential problems investigators may encounter when using these remote measures, and then how to anticipate and monitor them. And so in our discussion, we talked about how the problems encountered in multi-site trials are those encountered in single-site trials as well, but they're amplified with multi-site trials because of the additional variability introduced from the additional test administrators and potentially different types of technology in populations, micropopulations across sites. So in other words, inter-site differences may increase variability in multi-site trials, specifically in data elements that are important to the study. And these are the four kind of main themes that we discussed. So first and foremost, we spent a lot of time talking about the test administrators themselves. If you are able in a multi-site trial with remote assessments to have a central test administrator, that could greatly reduce the variability, but that's just not always possible, especially if your regions are global. So what we discussed was that it was important to standardize the requirements for qualifications, for training, retraining, et cetera. And so there should be a pre-specified plan that defines those requirements for qualifications, training, and even criteria for retraining. So there should be an effort to monitor the actions of these test administrators and retrain when applicable. Besides the passive component of training and reviewing qualifications, there could also be an active component where you would require, as part of the qualification, a certain threshold of agreement with what I should have said central rater here, with a central rater who is your own sort of studies gold standard, if you will, and that can be part of the active component of a qualification process to ensure that you're standardizing the training and confirming qualifications as much as possible across the various regions that you may be working. In terms of monitoring and identifying issues, course correcting, et cetera, we discussed the one potential solution is you could have a central overread of a small percentage of your data to really monitor the inter-rater reliability across your sites and course correct when applicable, when appropriate. So besides those were the obvious ones that we talked about right away, but then we also had a discussion about some more subtle effects that could still have a very large impact on your data. And that's the effect of the motivation and the buy-in of your test administrators, even if they're fully qualified and the training is standardized and everyone meets the requirements. If they're not motivated or feel accountability for the data, the integrity of the data could still suffer. So we talked about incorporating initiatives to engage the various sites and the various test administrators so they could have buy-in and accountability for the data. Okay, so moving on to the technology portion, I think this was covered in many of the panels and talks from yesterday and even this morning, but again, with multi-site trials, the variability across devices, internet connection, et cetera, may be even more amplified. So it is important when possible to standardize your mobile technology, but if the standardization is a barrier for either recruitment or an important goal of your study, then what you could do is standardize the high impact variables, and then everything else that's relevant, other relevant data elements as sources of variability should be captured in the data so that you have the ability to evaluate it, either statistical tools or otherwise in monitoring to ensure that it doesn't have an important impact on your data. Finally, in terms of technology, just because of the variability, especially across regions and internet connection and what they have access to, it would be best practice to, if you can, get a device that saves locally or even functions locally, so you're not dependent on an internet connection in order to do some of your assessments. If the internet connection is required because it's a live assessment, for example, you could consider providing devices, if possible, like a MiFi device or some other internet device that could standardize the connection speed for all of your participants. And then, so besides the administrators, the technology, the participants themselves in multi-site trials, this This is also very important. You may have different types of cohorts depending on how spread out your sites are. And so the cultural variability and norms, as well as other environmental factors like the size of their house that they're sitting in, what they have access to at their home. Do they have AC? Do they have heat? All of those environmental factors, again, are and cultural factors, cultural and environmental factors are amplified across sites. So it's critically important to collect the demographics and other common data elements so you can conduct subgroup analyses, so you have the ability to measure the impact of these variables on your trial. And then for the cognitive assessments themselves, when it is needed, make sure you have upfront the translation, back translations, cultural equivalence testing, so that based on what Dr. Pena just very, very thoroughly described, so that you can feel confident in the data that you collect from various cultural groups. And then number 4 here, this is less of a problem and more of a interesting topic that we discussed, which is the tension between standardization and naturalistic testing environments. You could overcorrect in one way or the other. And so ultimately what we discussed was it's important to consider the context of use when you're thinking about your prioritization. Do you want to, standardize everything or keep it more naturalistic? You should try to standardize variables that contribute to noise in your endpoints, specifically your primary endpoint, whenever possible. But it is possible also to overcorrect, so you should be aware of controlling for too many things and encountering collider bias, which can affect your interpretations of causal relationships between your measurements. Okay, thank you very much.
Fantastic. Thanks a million. I wanted to point one thing out, because I don't think that we mentioned it before, and that is that we have obviously a lot of academic contributions here, but Svetlana is actually from Neurocrine Biosciences, and they've done some very successful trials. So we also wanted to have intentionally brought in some company input as well. And so we really value obviously everyone's contributions, but it's great having, we're trying to sort of get a broad representation of interested parties. So thanks a million, Svetlana, and there will be some discussion. I know there's going to be discussion. And so our next group is breakout room 3 on safety and reporting issues. And we have Dr. Travis Shipley-Scott, who's joining us from group 3. Thank you.
Thank you. Let me just share my slide. All right, there we go. Yes, hi everyone. My name is Travis Shipley-Scott. I'm a clinical neuropsychologist, and our group mainly discussed safety and reporting issues. We did briefly touch on some validity issues, because I know the work groups there was, I think, more people than possible for that additional breakout room. But I'll focus our conversation mainly on the safety and reporting issues. That's the vast majority of the time that we spent, and I'll let the other work groups touch on the validity issues a little bit more. So the goal overall for our breakout room was to discuss safety and reporting issues that are unique to remotely administered cognitive measures, particularly with telehealth administration, so like our video telehealth assessments. And so we were really looking at a few different things. What are the safety issues maybe encountered when using remote administration methods? Do standard reporting mandates apply? And then also developing safety monitoring plans. So to summarize what we discussed, we kind of had 2 main overarching points with regard to these questions and issues. The first is planning is key in setting up your telehealth assessments. So, really just having a plan in place for how safety concerns will be handled prior to starting your study, prior to starting the project. This not only means understanding and knowing various, state mandates in terms of reporting issues. So knowing when and in what particular state, especially if you're doing things across state lines, what are the mandated reporting guidelines in that particular location, who you would contact if there is a concern, things like abuse and neglect or suicidality. Speaking of suicidality, having plans, having like structured questionnaires, things in your study in place to directly assess, whether or not a patient is at risk to themselves or other people. We also talked about, with regard to that as well, getting information that will help in a safety situation and in a situation where you need to work to make sure that the individual is safe. So getting relevant information from the person you're working with, the participant or patient, such as their address, alternative phone numbers, their phone number in case the call or contact gets disconnected, the maybe local hospital or other resources in their particular location. So if there is an emergency incident, what you can do immediately to respond to that situation, given that you're not in the same location as the person, and have that all ready prior, again, to the to starting your contact with that individual. And then in terms of working with our participants and patients, really setting expectations in advance. For instance, I think one person in our breakout room mentioned they in their kind of pre-visit, they inform the participants that make sure that they're dressing in a way that they're comfortable being seen, that they can't obviously conduct the assessment while driving, monitoring for things like substance use and so on, really monitoring the environment as well, beyond what the participant may tell you. And then feeling comfortable to set boundaries for when to possibly discontinue an encounter if needed. So if it seems like the person isn't in a safe place or in the right set of minds to do the visit, making sure that when it would be appropriate just to stop the visit altogether and reschedule. And then the second main theme that we talked about was really just needing to expect to encounter challenging situations, realizing that if your study has you're going back and reviewing data, and maybe you're not doing the encounter with the participant real time, but they're sending you something from a different time point, previous week or another day, having some sort of protocol in place to monitor for safety concerns, even if you're not meeting with the individual in real time. So this could be spot checks in terms of reviewing data to just make sure you're looking for any sort of safety concerns within the participant environment. Again, someone in our breakout group mentioned that in their particular study, I think they were doing EMA, and they were reviewing the records, and then they maybe noticed something that could be evidence of child abuse. So in having a protocol in place for just occasionally just checking randomly, even before you're doing the actual kind of a data collection piece and getting the data from the particular visit, just to make sure that there aren't ongoing safety concerns that you wouldn't have caught close to the time when the actual incident happened. So those were the main topics that we covered in our session. Thank you all for your time, and thank you.
Thank you very much. That was fantastic.
Green.
Yep. So our final presenter for the breakout group is going to be room 4, and this is equivalence versus validity reliability comparison between remote and in-person. And Dr. Alina Plant is going to present that. Dr. Plant.
Yes, unmuting is helpful. Okay, thank you. So we had a wide-ranging discussion, as you might imagine, and here are our takeaway points. So one of the things that came up early on was that face-to-face is not, or not all face-to-face measures can translate to a remote session, and we need to be okay with that. So in those situations, we may really be looking at developing new measures rather than adapting old measures. And when we're developing new measures, then they have to be validated and reliability established in the same way that a face-to-face measure would be developed. Okay, so the other related to that point is that sometimes when you do translate a face-to-face measure to a remote assessment, you may actually not be measuring the same construct that was originally intended. So you do have to look at whether or not that translation is adding a dimension that increases working memory load or changes the spatial rotational dimensions or things like that that could change the construct in a way that would interfere with your intended interpretation of that data. This has come up in the past, but it came up in our group again that gold standards can be flawed, and that I'm just going to put in here too that it's not only the gold standards can be flawed, but what the gold standard you're trying to link to needs to be linked to the way you're using that data. So we all tend to look at these through the lens of what we're doing and don't think about the range of ways that assessments are used and interpreted, and that a gold standard for one purpose is not the same as a gold standard for another. And that there was, I thought, a thoughtful expression in our group that we may need to create a new framework for how gold standards are established for remote assessment. Related to these other points is when an assessment fails, we should actually explore why it failed and to understand a better way forward in terms of developing new measures. So I thought that that was a really great point of consideration. And along those lines, input from stakeholders with varied backgrounds is important for all steps of the validation process. And so there was a great example given where there were psychologists and engineers in the room together, and that their perspectives on the problem were very, very different. And it was the combination that actually resulted in a better product. So we also heard a really good example already this morning about needing, in kind of a pre-development step, needing to get the perspective and user experience, desired user experience from the end users into consideration and into the mix before you even advance to the validation and reliability work that you might need to establish for your measure. There was discussion around there was a tension between standardization and related reliability, because one of the reasons why we standardize things is to increase reliability within the study, and then to increase reproducibility across studies. So there is a tension between reliability and standardization and inclusion of the breadth of the population. So the idea that if we have a measure, but only a subset with a certain kinds of motor skills or visual acuity or hearing levels or whatever it might be, can actually use this tool, you get a more homogeneous sample, but you get less representative samples of the population of interest. So that needs to be carefully considered. And then finally, a suggestion for something that would actually be helpful in terms of equivalence and reliability and validity, and that is that as people are developing data responsibilities, it would be really great to have some overlapping measures across these data repositories that would help us establish concurrent validity and internal stability data, particularly for disorders that are rare in the population, where any one study might not have a big enough sample to really get robust measures on these things. So thank you.
Fantastic. Boy, it's just, these are just phenomenal. If we could please get all 4 breakout leaders. And while we're doing that, I also wanted to just acknowledge each of these breakout rooms also had an NIH partner with them. And so in room 1, we had Dr. Holly Storkel. Room 3, we had Dr. Rebecca Hammer. And in room 4, we had Dr. Tracy King. So thanks to all of you for engaging in this and being a part of the effort. All are also members of the planning committee, as Christy mentioned yesterday. So boy, there's so much to talk about. So one thing that I think I wanted to start with is, because it goes a little bit to the validity question, sort of, as a larger topic, and that is, how do we define the one thing that I haven't heard a lot about? Someone mentioned it yesterday, but I think it bears on the topic in at least 2 and maybe 3 of the rooms, is how do we define meaningful clinical change? We keep talking about this gold standard thing, and we also heard yesterday from Anna Williams about her experience with her daughter. How do we define meaningful clinical change? How does that work into all of this? And I'll let anyone take that first. We're going to sort of go around the room. Anyone?
I can start off. So we do this. I also do treatment research, and so this is a topic that we think a lot about. And basically, it's one of those things that if it's meaningful to the patient, or meaningful to the parent, or meaningful to how much change would be worth it to you for the investment of time, then that's meaningful change. So it's sort of like how much change depends on how much, like, you can have a very small amount of change, but it has a big life benefit. So that might be meaningful change. But in other domains, it may be, okay, I've spent, how long am I willing to stay on this behavioral treatment before I switch to something else that might work better? You might have a bigger standard for a bigger amount of change for that. So there is no like one standard across all conditions. It really is what's meaningful.
I totally agree. And as someone who works mostly on interventional trials, we think about this all the time. And when we set up our statistical analysis plans, they're mostly focused on detecting statistical change, which, if you include enough subjects, you can usually get there. But then the question is always what's meaningful, not just what's statistically different. Did you meet your pre-specified P, but what's actually meaningful? And I think specifically for interventions, you have to take into account the risk as well, because there's no drug or device that comes without risk. So it has to be balanced by that. And the way that we tend to answer this is by infusing the patient voice, which Elena just mentioned. We ask the patients, we do surveys. You have to infuse that component into it. We also ask the clinicians themselves, who are very familiar with, each specific indication and group of patients. But I think that that's a critically important question, because even for behavioral interventions, you might wonder, well, there's not, a safety risk, but there is a time component for children with, autism or other behavioral disorders. They may, it's not nothing for them to have to schlep over somewhere or be in front of a screen. So I think it is important up front to define what is that delta that's meaningful in an intervention. And it may be a smaller one for behavioral ones. It may be a larger one to take a drug or device. But yeah, I think the way that you get to that is infusing the patient voice and asking them directly.
Liz, you look like you want to say something.
No, I'm just agreeing with
Oh, okay.
the comments. Yeah, I mean, what is the impact? What's a difference that makes a difference for the patients that we're working with? I think is the difference what we want to try to define, and it's a little bit of a moving target, but I think but I think it's doable.
Sorry.
Travis, did you want to jump in?
No, I mean, I agree with what everyone says, and it's interesting. I just was looking in the comments here. And my work with patients with dementia, yeah, definitely in neurodegenerative diseases, stability, definitely can be a goal. You may not have, obviously, as we know, there's no reverse engineering, massive cognitive improvement in patients with dementia, but just seeing stability or preventing future decline, I think can be really, really important as well. So I definitely agree with the comment there in the chat.
So let me ask this only because I know the person who wrote that. That's Nicole Farhat, who's one of our clinical program research managers in the PEDS group at NINDS. And so Nicole actually worked with children who had Niemann-Pick type C disease. And so as I'm thinking about it, some of the neurodegenerative diseases across the age spectrum have medical complications superimposed on that underlying disease. So as an example, epilepsy, I'm an epileptologist, so that's the example I'm going to use. So epilepsy can be a coexisting disease process, again, across the age spectrum. And sometimes we're looking for the impact of, so you already have this either, underlying neurodegenerative disease or genetic condition, pick your favorite disease. And on top of that, you have something else. So how do we approach from a validity perspective, from getting some sort of a norm, and I'm putting that in air quotes, intentionally. How do we separate the associated diseases, medical complications, if you will, from the underlying disease when we're thinking about all of these issues about validity and how we're going to, best actually access what that person's abilities really are? Kind of a complicated question, but it's one that I think about all the time in my epilepsy clinic, because we're trying to sort out, is it the medication, is it the seizures, is it the underlying, like how are we doing this? Anyone?
I think your comment is a common is one of the especially in epilepsy trials, one of the very common things that people bring up, where in the trial there is a historical endpoint that's been very successful for much of the drug development, and that's counting seizures. What is the seizure frequency? And what we hear over and over in all of my rare disease trials, especially the pediatric epilepsy trials, is that for many of the pediatric epilepsies, it's not always the seizures. There's many DEEs where the seizures are not even the main complaint. It's all of the different comorbidities. And all I can say is I think we need to measure them. We need to look at them. We need to measure them. But from the sponsor perspective, where we come from is we understand they're important and we want to include them in our trials. But the endpoints are so difficult to measure in a meaningful and changeable way over the course of a 3-month placebo-controlled trial. And so when we want to include them, but many of the cognitive endpoints take much longer than 3 months to change. And no child is going to be in a placebo-controlled trial for 6 months to a year. I mean, then you start talking about ethics. So I do think we're in between this rock and a hard place, but the more we talk about it and the more we agree that it's important, then there's an innovation workshops like this where we can discuss how do you best measure these other comorbidities. And then if their measurements are there, they're going to start getting incorporated in the studies.
I'm going to take a different tact as somebody who mostly works at the behavioral end, and that is sometimes we don't care. So, we know that certain disorders come with, multiple comorbidities. And what we really care is, like, who does this treatment work for, and how often does it work, and by what magnitude does it work? And then you can start saying, okay, well, what about the kids with developmental language disorder who also have ADHD? Or what about the kids with autism who often also have the seizures, and start looking at subgroups. But if you're not looking at medical interventions that really do, or drug interactions might be a concern or things like that, you might not care.
Other thoughts? Okay, so here's a question. This one's a little more related to, this is a little more brass tacks, and this comes from Dr. Rebecca Hammer, who I think was in room 3. Some of the investigators mentioned that they face challenges in study section to review and proposing to use remote assessments in their research. This might differ across disciplines and patient populations. Any tips or hints on how to manage study sections? I mean, it is people say NIH said whatever, but it's actually your peers that are saying it, peer review. So thoughts about how to handle some of these issues when you're writing a grant application. Anyone?
I don't know. It's really hard. I mean, I always try to put myself in the reviewer's shoes, right? So how do you mitigate potential objections to your strategy, and then you try to alleviate those concerns? I mean, it's really a general issue, no matter what it is you're doing, that there's going to be questions about the validity and the reliability of those. And even when you don't need pilot data, piloting is going to be really critical to address potential concerns. There's no perfect way. And yeah, to me, I don't know. I don't have an answer.
So one of the comments from our keynote speaker yesterday in row column, grant writers need to know the literature and have experience with remote assessment in order for a viable proposal. And so it's an interesting.
I'm actually going to disagree with that. And the reason why is, first of all, good luck with that. Getting that narrow band of expertise on your review panel. But basically, reviewers know what you tell them. And so if they need to know something about remote evaluation in order to appreciate the rigor and reproducibility that you've built in, then tell them.
Great. Other thoughts on that? Okay. Moving to another question. So this comes from another planning committee member, Dr. Holly Storkel. So multiple groups talked about not being able to adapt some measures, and it may be best to start from scratch. Do you have thoughts, or what are your thoughts? I know you have thoughts. What are your thoughts about what the characteristics might be for tools where adaptation to online might work versus tools, purposes, functions where it may be best to start from scratch? So adaptation versus starting from scratch.
I think it depends on what the question is, right? What is it that we're trying to test? What is it what data are we trying to collect? And what is the technology bias? So I try to think about technology and remote testing in terms of what can this bias that doesn't that humans can't do easily. Right. I mean, that's the beauty and the promise of using technology. So, one model that we've used is kind of like, do we want to replicate what we can do humanly in terms of face-to-face testing? Do we want to or do we want to transform it in some way? And so, one of the this is just a really small point, but one of the things that we've been working with, and a lot of people have been working, is with speech recognition kind of technology. So it's really good at some things and it's really terrible at other things. And so maybe we just need to maximize what it's good at and don't worry about what it's not good at, so that we can harness like the benefit more maximally and not worry about the stuff that it doesn't, or it can't, or never will do.
So in our group, the issue of change of construct came up. So, and there are lots and lots of ways there where this can happen, particularly if you're doing a bring your own device kind of a scenario. So for example, if you've ever given the Kaufman triangle subtest where kids look at an image and then have to make the triangles here. If people are doing that on their own device and their device is up like this, probably no problem. But if they're using their own tablet and their tablet is down on the table in the same plane of rotation where the triangles are, now you've got a different construct because you don't have that flip in spatial rotation anymore. The same thing can happen with the token test where there are tokens to be manipulated. So I think you need to really think carefully about how the end user is going to experience the assessment and whether or not it actually is replicating what is happening face to face. And if it's not, then you then need to go to step 2 and say, okay, are these differences actually going to correspond to differences in processing that will change what we're measuring?
Yeah, I've been thinking about this issue for a while now, and I keep going back to some of the things that were said yesterday. You know, as a neuropsychologist, thinking about the current measures we use and the history of them, and how, you know, a lot of them obviously very, you know, much predated the use of technology, you know, based on decades old paper and pencil measures. And thinking about Dr. Soy's talk yesterday about the TabCat and, you know, these newer digital measures, and, you know, kind of thinking of some of the maybe advantages in terms of even taking it a step further in terms of like ecological validity and you know, how these measures could map out to, you know, real world function versus, again, how our neuropsych tests were originally developed as well, with you know, the idea being mapping onto certain brain functions, and, you know, ideally, you know, combining those in some way such that, you know, you can, I think this maybe was brought up a little bit in our session yesterday, you know, considering newer remote assessments developed digitally compared to gold standard measures developed paper and pencil, you know, having the advantage of, you know, mapping on to our prior neuropsych tests, but also maybe being a better in terms of actually mapping out to real world functions similar to things we do in real life, you know. I find that very interesting and possible direction. I really enjoy the new developments on these, you know, digital assessments that are being created, designed to be administered remotely, that, you know, still really map out well to our cognitive functions. I think that's maybe an ideal direction to go, you know, in the future.
Other thoughts? So another question is, how do you think about incorporating common data elements into these remote assessments? I know we brought it up yesterday in our group. It was a very specific thing that we were talking about, but just thinking about it a little bit more broadly. What role do you think common data elements can play in how you're designing the studies, and also down the road when you're trying to, you know, put a bunch of studies together, finding some different studies back together again?
When I summarized it for breakout 2, the purpose was so that you can, in a standardized way that can be compared across sites, but also across other trials, define the population that you've enrolled and be able to do subgroup analyses in a more standardized way. So I think there are some data elements that should just be incorporated in every trial, right? That is not age, sex, and for cognitive educational attainment. I mean, there should just be a standard set of things that define the population that you've enrolled, and that make your trial able to be understood in the context of other trials of a similar topic or of a similar intervention. And one of the main things you need to be able to interpret it, especially in a publication, is who did you enroll and what did you give them? What intervention or measurement did you assess? So I think it's just about having enough information for a reviewer to understand your interpretations from the data.
Other thoughts? Thank you. Other thoughts?
I would add linguistic, language knowledge, right? I mean, half of our world is bilingual. There are a lot of other languages spoken and we ignore it to a large extent. So I think that we need to understand like the linguistic diversity of our populations in a much more nuanced way. And it may not even be bilingual, not monolingual, but I think that monolingualism is often thought of as the standard and the norm. In the US, a third of our kids have exposure to other languages. And I don't think that that is something that we can ignore. And we need to start asking these questions in a more systematic way.
So one of the follow-up questions was, what specific elements might be important specifically for remote assessments?
Okay, as the validity and reliability team person, I'm going to point out that one of the chat people said, should we code location or device? And from a psychometrics point of view, it would sure be helpful if people would do that, because there are real questions about, whether screen size matters or whether, orientation of the device matters or whether they were home with 3 other kids who were running through the background. I've certainly seen that. So those things really would be helpful to start determining under what circumstances can we get reliable and valid data from these measures.
Yeah, I'll just say I tend to think of this a little bit as the chicken and the egg, because in the summary from breakout 2, what I said was, consider the impact of those various variables and try to standardize the ones that are high impact. Sometimes, you can't. There is a trade-off between how much data you collect and then how tired the people get, right? And everyone has to work with that balance. And so what I had said is standardize, collect the high impact variables, and then the others just do your best, right? You can only do so much, but how do you know what's the high impact? And that again is data collection. So, I think as remote assessments become more common and there's more research done on them and the validation of them may become more standardized, this I hope that this becomes something that is part of the studies of these assessments. What are the high impact variables? And so just to answer, from my perspective, the question, what elements are important? What should I include? I would think about, what do you think for your endpoint, what you're measuring is going to introduce the most variability, is going to have the most impact to your outcome, and try to collect that. And then the other things, you may have to do in another study, or you may have to rely on the literature of what other people have measured, because no one study can capture everything. So you should try to prioritize based on impact.
So, so something that's related to this is that, and this sort of gets to that last point that we were making in our group yesterday, Svetlana, about, the naturalistic environment versus the standardization part. And one of the things, so that's actually part of how this all rolls in. And so there were a couple of comments in the chat, and I'm just, I guess, more looking for reaction more than anything else. The question is, should remote assessment designs also include data elements describing context such as devices and location? And then another comment related to that is recording environmental factors such as ambient noise and internet connection speed when applicable could be useful as well. Thoughts about that?
We talked about that in our breakout as well, where the easier you can make this, like if you give them a website for a speed test and you say, okay, click on this link and take a screenshot, and you could sort of guide them quickly through something, because someone in our group said that she tries to do it and that the participants will get confused and flustered. They don't even know where on their computer to look up what type of device they have. So I think some of this could be thoughtful up front, right? You can ask them maybe before your session in a pre-assessment form to fill out these things and maybe give them pictures of where to find it. So it's not part of the assessment. You don't get the fatigue right before you do it. But again, the more you ask, the lower the compliance with your study.
Okay, I'm going to point out that to a psychometrician, everything's a measurement problem. So, you can get a variable on anything and measure its impact. But I think that I'm going to agree with Svetlana, and that is that we probably don't have the tolerance to measure everything. So we're going to have to pick our battles here. But if you're dealing with low incidence populations, small, collections of small randomized clinical trial type studies, having some collection of standard variables would allow for aggregation of the data that would allow issues to be addressed that are beyond an individual lab to do.
So a related comment on this. This comes from Bridget Kelleher from Purdue, who presented a data blast and had a talk yesterday. She said, yes, in clinics, community clinics in particular, are not immune to variability in testing variables that can impact performance. The degree to which variables are impactful will also vary across populations and individuals. These issues are not new, although, of course, become more salient in remote contexts. But it is something, we've all been in testing rooms that are a little colder one day than another. There's construction noise or, all sorts of distracting things. A light bulb or two are out, and we've all experienced that. It's a nuisance for us working, but it does, maybe we need to think about that a little bit more. One of another comment that came up would be good to hear more about passive data that could be collected or already available on various devices, but may not be accessed or used. Also barriers and needs here, and this is sort of related to CDE. So passive data collection.
I think if you're using the same device, if you're able to send the same device to all the participants, you could do that, and then you have less of an issue with variability anyway, though, if you have that, because it's always the same device. I think it's hard to get the passive data collection if they're using their own computer.
Any other thoughts on that?
Yeah, so certainly we have, passive recording devices like Lena has come up multiple times. I'm going to point out that if you're recording by Zoom, you now have an audio record that, as a speech and hearing person, I could analyze in terms of ambient noise. But I also want to mention that the way I think about data collection is like what has to come from the subject, and then fatigue becomes an issue. But what information can I get from other people? So there are lots of, parent report devices that might be useful for my study that doesn't cost my subject time. So they're not quite passive, but they don't tax the subject anymore. And so you can think about, where the balance is between what I ask directly from my subject and what I get from other sources. For children, that also might be like schoolwork, writing samples or whatever it is. Yeah.
Mm-hmm. And Elena, could you collect some of that before the actual time of assessment?
Oh yeah.
Like for example, when I have little children, and when I take them for their annual visits, I get this long questionnaire the week before I go, and it takes forever to fill out. But then when I get there, we don't talk about it. We do the measurement.
Yeah, absolutely. Yeah.
Interesting. So the one of the comments I'll just add, this comes from the NINDS CDE leader, Dr. Carolina Mendoza-Puccini. She said, agreed, you need a core set of elements. The challenge is to agree on that set of elements. The core elements are usually a short list of CDEs that aren't burdensome to collect. And so it sort of relates a little bit to this discussion. The other comment is, in some discussions our team has had with Fitbit for wearables, there are questions about how long the data can stay on a device, whether high frequency data erases after days automatically due to bandwidth issues. Do similar constraints apply for neuropsychological testing and the devices that are used to collect those data?
I haven't run into those issues. It's not usually, I see the others shaking heads also. They're not usually such high frequency data. They're like questionnaires. And these days the devices are really good. And in my studies, which are large global studies, the devices can sit with the data kind of indefinitely. I've had issues where they only need an internet connection to sync it to the server, right? It stores locally. And I've been in situations where for whatever reason, it didn't work. And so even in those cases, they could ship the device to the vendor and the vendor gets it off and puts it in the server. So even like 5 years ago, it wasn't that good, but now data loss has not been a huge issue for us, at least.
Anyone else?
Yeah,
So
I haven't seen this either, you know, in as a neuropsychologist, and the studies that I've been in that have used like tablet-based assessments and things like that. No, I mean, I don't. I'm trying to think of an example of data that's super high frequency like you would find in a Fitbit. I haven't encountered it yet. So.
Great. So one last comment, we're currently collecting normative data in both English and Spanish on our new comprehensive tablet-based neuropsych battery, CCAB, that we deliver to patients' homes that includes internal cellular connectivity so everyone can participate. This is funded by an NIA SBIR funding mechanism. They have 900 participants so far. Examiners can see and hear examinee remotely and make timestamped notes of any background noise or distraction. Examiner monitors ongoing performance online as well. So we have a number of, I'll just point this out to everyone as we're wrapping up. We're going to wrap up a couple minutes early, give you some of that time back that we took yesterday. So a number of interesting papers and abstracts and proceedings have links in the chat. Please look at them. This is part of the whole information sharing exercise, part of a workshop like this. And so I know we have, and we've been consistently today, I don't know if anyone's noticed, we've been consistently around 100 people. So it's not exactly the coziest environment, but that chat room, the chat really has allowed us to connect up in ways that I think have been extremely useful for everyone. And so, so I wanted to thank our breakout leaders. All of you in your groups did an absolutely phenomenal job. Thank you so much. Thank you for engaging in the discussion. It's really, I mean, I've learned a ton. There is going to be an NIA, NINDS workshop on clinically meaningful outcomes that Christy posted in the chat. So please take a look at that too. It's going to be in March, a workshop. So, you know, there, as you can see, there's an interest on the behalf of NIH in this area. We want to really try to, you know, lift all boats, make things methodologically, improve the methodology, and hopefully make things more relevant, obviously, to the people who are most impacted by these diseases, illnesses, and disorders. So with that, we're going to go to our noon break, and we're going to come back at 12:30. And at 12:30, we're going to have 2 talks and a panel discussion. We're going to have Dr. Catherine Papp is going to be talking about novel and emerging methodologies and proposed framework for developing standards. And then Dr. Shefali Singh is also going to be providing us with a talk, and her topic is accessible design. And so, and then we'll have a little bit of discussion, and then we have another set of breakouts, and then we'll be wrapping things up for the day. We should be bringing the plane in at 4 o'clock. And so thank you all once again. We'll see you back at 12:30 and we'll have some more talks and discussion. Thanks very much and have some good time off. Welcome back, everyone. We're gonna wait a minute or two. And then we will start. Just like we had great content for you in your postprandial state yesterday, I think we're going to have just as good content this afternoon in your postprandial state. Or at least I hope you had something to eat or refresh. A friendly reminder, so I'm getting my COVID vaccine later today, and I just want to remind everyone to get your flu shots and get your COVID vaccines. We don't want to see you in the hospital. We want to see you walking down the street or on the street. So please take care of yourself. All right. I think, are we ready to launch the videos? I'll do the introductions.
We're ready.
Fantastic. So it's a real pleasure for me to introduce our next two speakers. We'll do the introductions and then we'll run their videos. Like I said, the content, I've seen both of these talks already and they're both fantastic. So Dr. Catherine Papp is on the faculty as an assistant professor at Harvard Medical School. She's also a clinical neuropsychologist at Brigham and Women's Hospital. Her clinical practice focuses on the assessment of memory disorders, and her research, as you'll hear, involves early detection of cognitive decline in Alzheimer's disease and related dementias using digital technology. Our next speaker is Dr. Shefali Singh. She's the founder of Cambridge Neuropsychology. She's the director of the Digital Cognitive Research Program at the Laboratory for Brain and Cognitive Health at the Institute for Technology and Psychiatry at McLean Hospital, also at Harvard Medical School, and she has a lecture appointment at Harvard University. So with that, these will be great talks. Let's go to the videotape.
Good afternoon. It's a pleasure to be able to speak with you today. My name is Kate Papp, and I'm a neuropsychologist at Mass General Brigham. I'd like to provide an overview of current thinking and approaches to digital neuropsychological assessment. This is a field which has grown exponentially in the past few years in the context of COVID. And while there's a lot of exciting novel approaches, there's also important standards that must be met before these digital cognitive assessments undergo widespread adoption and implementation, particularly as it relates to their use in clinical settings. That said, while I'll briefly discuss using digital cognitive assessments in the clinic, the focus of the talk today is really on applying standards and methods for digital cognitive testing in the context of research. Here are my disclosures. First, I'd like to speak about the definition of remote neuropsychological assessments. Second, we'll discuss some of the advantages as well as the disadvantages of this approach. Then I'll talk about the standards for ensuring that remote neuropsychological testing is feasible and valid. Next, I'd like to talk about research applications for remote neuropsychological assessments using early detection and tracking in preclinical Alzheimer's disease as a model. And then finally, I'd like to share some of my own work on the development of the Boston Remote Assessment for Neurocognitive Health, which demonstrates carrying out feasibility and validation steps in the real world. So first, I'd like to talk about how we can define remote neuropsychological assessments and distinguish them from other approaches such as teleneuropsychology and passive monitoring. So by definition, remote neuropsych assessments are completed outside of the clinic. They're unsupervised. There's no rater or clinician guiding the individual through testing. Testing can be completed on either personal devices or those provided by researchers. And while there are specific types of hardware that can be used, such as digital pens or eye tracking devices, the most common approach, and what I'll be focusing on today, is testing completed either on smartphones or desktops via either native applications or the web. And then finally, remote neuropsych assessments are task-oriented. That is, the participant knows they're being assessed and they complete a series of tasks. Now, these differ from teleneuropsychology, which offers a standard assessment via video teleconferencing with a rater or clinician administering the tests And this is a procedure that's become relatively widespread in recent years, and it's just another aspect of clinical practice. It also differs from passive monitoring, which takes a big data and agnostic approach to collecting lots of different types of data from an individual's usage of their smartphone, to their navigation via GPS, to other biometrics like heart rate. And then also which attempts to mine that data for disease relevant signal. These are fields, teleneuropsychology and passive monitoring in and of themselves, and are too much to cover in our time today. There are several advantages as well as disadvantages to remote cognitive testing that are important to consider. So first, by being able to be completed remotely without a visit to the clinic, these types of assessments are by definition more accessible. They may allow research participation among those who are geographically remote or who are unable to travel to research sites for other reasons, which can enhance equity and inclusion. Because they're unsupervised, a remote assessment doesn't require the time and cost involved in having a rater or a clinician work with a testee one-on-one. And then because they're both remote as well as unsupervised, this really offers the opportunity for scalability. That is, much larger samples are able to be collected than would be achievable with in-clinic testing. And this bodes very well for genomic studies, epidemiological studies, as well as studies in asymptomatic stages of disease, such as the case of preclinical Alzheimer's disease, which I'll discuss later. And while neuropsych assessments by definition use standard administration and scoring, the level of precision in standardization in digital assessments is much greater. For example, we have precise control over how long a stimuli is displayed to the millisecond measurement of response time. Now, regarding disadvantages, a major one is data fidelity. Because they're supervised and remote, can we ensure that the person who we think is taking the test? And also, are they doing so without the use of memory aids or a family or friend who's helping them do the testing? There are a number of formal procedures that can be put in place, collecting biometrics like a fingerprint or voice. These are expensive and sometimes only work better in things like native apps versus the web. But it's an important issue that we have to consider about whether we're capturing data from the individual without cheating or aids. Next, a disadvantage is that this testing is occurring in the wild. It's an uncontrolled test environment. We can't control for interruptions from outside, interruptions potentially related to technical issues. And so that in that way, data can be more messy because it is a unstructured testing environment. There's also technical problems, from difficulties at the level of the user to difficulties transferring data from a device to the cloud. There's also a lot of contingencies that need to be considered in the wild as regards to technical difficulties, with participants starting and abandoning sessions, getting frozen out, dealing with partial or incomplete or duplicate administrations. And then in technical difficulties, we hear terrible stories about how data is collected at the individual level, and then there's some disruption between when it's collected there versus when it gets to the cloud. So there's also the issue of potentially having missing data. There's also known variability across devices and operating systems. For example, reaction time across different smartphone platforms and operating systems differs. There's also variability depending on the web connections and how strong or weak they are that can impact the precision of measurement, particularly as it relates to some of these reaction time measures. And then another consideration is test security, that we're sending out our tests. People can feel free to take screenshots or copy things down. And then we have to wonder how that will bode for future studies that are using the same types of tests and forms. And then finally, a perennial question is around digital literacy or user acceptability. So there's really positive trends in the US, such that 61% of older adults over the age of 65 had smartphones in 2021, and that number is growing annually. There's also some evidence that the usage of smartphones is pretty comparable across different demographic groups, including Hispanics, whites, and Blacks and African Americans, which bodes well for equity. But there is still a potential for people who are less familiar with digital devices or less willing to do testing in this type of format. Now I'd like to talk about what standards a remote neuropsychological assessment should meet. So as far as I'm aware, there's no current consensus on standard practice for the development and use of digital cognitive assessments. However, I think most of the standards of general cognitive test development apply to the development of these digital measures. So that said, there's a few that are much more pertinent to digital testing, including data fidelity and user acceptability. I already spoke about data fidelity and the importance of ensuring that the data that's being collected is completed by the participant without the use of outside help. User acceptability refers to an individual's willingness to take the task. Because there's no examiner who's guiding a participant through the assessment, it's really critical that the task instructions are clear, that the task difficulty is appropriate, that participants don't get either bored or overwhelmed based on the tasks being too easy or too hard, and that the amount of time required to do the tasks is reasonable. There's a lot of user acceptability testing, or UAT, that needs to occur prior to the implementation of a digital test. This is often a very iterative process with multiple versions and adjustments made in the context of participant feedback, from the size of font to the clarity of instructions to the length of the tasks and everything in between. Next, it's important to ensure that tasks are feasible, that they're able to be completed in their entirety, that they're valid, that they're measuring the construct or the criterion of interest, and finally, that they're reliable, that there's a consistency in measurement. Now I'd like to talk about the case for early detection and tracking in preclinical Alzheimer's disease, which is a use case which really highlights the potential utility in the future of using remote digital testing. So for those of you who are less familiar with Alzheimer's disease, I'd like to level set here. AD involves a long asymptomatic phase, starting with the accumulation of amyloid and tau, followed by subtle yet increasingly persistent cognitive decline. And our ability to observe abnormal AD biomarkers in living individuals using both PET imaging and cerebral spinal fluid, but now more recently plasma markers, has led to early detection during the preclinical phase of disease, as well as the implementation of secondary prevention trials, which try to prevent cognitive decline amongst asymptomatic individuals, so AKA among those with preclinical AD. However, capturing initial evidence of subtle cognitive decline, which is important for determining the efficacy of any sort of intervention or treatment, is really challenging. At the cross-section, as you can see on the graph on the right here, most cognitive tests are not related to these AD biomarkers, and declines can only really reliably be observed after 3 or 4 years of follow-up using testing that is administered in the clinic over relatively long intervals of every 6 or 12 months. So because of this, determining the efficacy of an intervention during preclinical AD is a long and expensive process, with studies lasting 4 or more years and large sample sizes of 1,000 or more, with many more individuals who are screened for inclusion in these studies. Digital measures really have the potential to address some of the challenges to assessing subtle cognitive changes not able to be overcome with traditional paper and pencil measures in the context of preclinical AD. This is a recent figure from a recent review by Oman et al., and it's helped to provide a lay of the land of digital measures that are specifically designed to capture cognitive changes that occur at the preclinical stages of AD. These digital cognitive tests and their platforms vary in their hardware. Some are cognitive apps, some are web-based, others use devices like digital pens, eye tracking, and virtual reality. There are also a number of different approaches to improving early detection. So these are some examples. The first is improving reliability through momentary or burst testing. The second is harnessing the potential of big data collection. The third is targeting cognitive processes that are more specific to preclinical AD. And then the fourth, which I'll be focusing on for the remainder of this presentation, is using serial assessments to capture multi-day learning curves. But first, I'd like to briefly discuss these first 3 approaches. And before I move on, I'd like to also note that this is not an exhaustive list by any means of these different types of early detection approaches. There's many different strategies, from adaptive testing to gamification, just to name a few additional ones. So in momentary or burst testing, participants are administered a series of brief tests multiple times per day on their own devices. For example, they may get 3 different tests administered 4 times per day for a few days. And then by averaging performance over multiple assessment time points and tests, the hope is that we would obtain a more reliable or less noisy measure of an individual's cognition. And you can see a visualization of this on the top right. So by decreasing the noise in an assessment, we can then be better able to more sensitively capture change over time. And this is really achievable by allowing for repeated testing with digital measures administered multiple times per day. Another approach to improving early detection is by harnessing big data. This is an example of using a digital pen, but the same approach can be applied to other types of big data, such as speech samples. Here, participants are asked to complete a familiar neurological task, clock drawing, but they use a digital pen, which captures hundreds of variables related to performance, such as pen velocity, pen lifts, thinking time, and then thousands of clock drawings have been collected in patients, and algorithms using big data have been developed to see what aspects of performance are associated with a clinical diagnosis. And then these algorithms can then be applied to new data that's coming in. And not unsurprisingly, a digitally scored clock versus a traditional hand-scored clock is a much better able to discriminate between individuals who are normal versus those who have MCI, as shown in the graph on the right. Another approach to improving detection with digital measures is to target cognitive processes more specific to preclinical ID using tasks borrowed from the cognitive neuroscience literature. There's examples here of specific types of memory changes supported by medial temporal lobe and thought to be vulnerable in preclinical ID. Harnessing these types of paradigms may be useful in developing digital tests that are more specific to changes in the early AD trajectory, with an example on the bottom right here of a pattern separation task. The last approach to digital cognitive assessment is the one I'd like to focus on for the remaining time, and this is using serial assessments to capture multi-day learning curves, and it's what motivated us to develop the Boston Remote Assessment for Neurocognitive Health, or BRANCH, as we call it. So I'll provide a bit of a rationale for this approach. So practice effects are the natural improvement in performance that we observe when someone is retested on the same measures. They've long been considered an obstacle that obscures the measurement of cognitive decline. On the flip side, there's been ongoing interest in harnessing practice effects to improve signal detection, given that individuals with preclinical AD exhibit reduced practice effects in the setting of completely otherwise normal cognitive performance. And also, these reduced practice effects have been shown to be associated with increased risk of future cognitive decline. Interestingly, recent evidence suggests that reduced practice effects in preclinical AD may actually reflect very early failures in memory consolidation. That is the process where a temporary memory is transformed into a more stable or lasting form, a process that requires the passage of time that's not afforded with a single testing session, but which is achievable with repeated digital testing. So for example, in this study, we gave 94 cognitively normal older adults iPads to take home for a year. They completed monthly testing using the Cogstate platform. We had them complete 2 versions of a face name memory test each month. In one, they had to learn and remember the same faces each month. And in another, they saw alternate or new faces each month. You can see that performance only diverged over time between amyloid positive and negative participants when they saw the same version each month on the top versus new versions on the bottom. So this finding of a reduced learning curves is that it's primarily observed when we have the same versus alternate content suggests that individuals with AD pathology are not benefiting from repeated experiences. These initial findings were very intriguing because not only is this type of memory paradigm particularly sensitive to preclinical AD, but it seems like it's increasingly feasible to administer using digital cognitive assessments and to potentially detect these effects over shorter time intervals, such as over the course of a week. With this in mind, we developed BRANCH, which is web-based cognitive testing. In addition to the face name associated memory exam, or FNAME, we also administered another associated memory test, the groceries test, where participants must remember a grocery paired with a price, and then the digit signs test, which is a measure of speeded processing modeled on the digit symbol substitution test. We had participants do these tasks with the same stimuli each day for a total of 7 days. We were interested in, first, are these tasks feasible, valid, and reliable among older adults? Second, does the multi-day learning curve differentiate between amyloid positive and negative participants? And third, is a less steep multi-day learning curve over 7 days associated with greater risk of future cognitive decline? So we had a total of 164 clinically unimpaired older adults recruited from the Harvard Aging Brain Study and other related observational studies complete the multi-day learning curves. 36 of these individuals shown on the left were amyloid positive and 128 were classified as amyloid negative. participants underwent in-clinic paper and pencil cognitive testing, and a PAC-5 composite score was calculated for each individual. And then a subset of 123 participants had follow-up longitudinal PAC testing. We took a number of steps to ensure that participants would complete the task each day. So this is encouraging feasibility a priori. We gave participants increasing compensation for each completed assessment. We kept it brief. There was a range of task difficulty. Some were challenging, others were more achievable. And we had fun brain facts were provided each day upon task completion. This is some initial feasibility data. So participants required an average of 12.2 minutes to complete the task. More than 97% of participants completed all 7 days of testing, which was great. Self-reported task enjoyability actually increased over days, as you can see from the graph on the right. People liked the test more and more each day. Technical difficulties were relatively infrequent, with the most common self-reported problems being poor device response to finger taps. And then regarding data fidelity, we asked participants to complete the tasks independently and without the aids for the benefit of research progress, and we asked them to attest to this. To get a sense of whether we were capturing participants' true cognitive performance on their own device in a remote setting, we looked at simple correlations between day 1 on branch and a participant's in-clinic testing on this composite, the PAC. And we found a moderate correlation, suggesting that we're really capturing valid information about cognition. In other words, this finding can be considered evidence of construct-related validity, and that our digital cognitive attest is associated with gold standard performance on paper and pencil measures in the clinic. I won't go into detail here, but we also looked at retest reliability of the multi-day learning curve, as well as concordance of alternate forms, so that we can track these learning curves longitudinally. So next, I'll discuss whether these learning curves were related to amyloid status and whether they predicted future cognitive decline. So to level set, we first wanted to see whether standard cognitive measures differentiated between amyloid positive and amyloid negative groups. We didn't observe associations between amyloid status and cognitive performance on these standard measures, as you can see in red. Also, we did not observe associations between amyloid and cognitive performance on day 1 of the multi-day learning curve. However, we did observe that amyloid positive participants showed a diminished curve after about 4 days of testing relative to amyloid negatives, with the strongest effects observed on the digit signs test, followed by groceries and then the phoneme, but a comparable pattern across the tests. This finding could be considered evidence for criterion-related validity, and that our digital cognitive paradigm is associated with a disease state of interest, in this case, biomarker evidence of preclinical AD. Another aspect of criterion-related validity with this paradigm is showing that lower performance on this digital measure is associated with a progressive cognitive decline observed when an individual is on the Alzheimer's disease continuum. So to this end, we examined whether an initially diminished learning curve, summarized as a single score on the x-axis here, was associated with longitudinal decline on standard in-clinic measures. And we found that, in fact, initially reduced learning was associated with subsequent decline on the PAC over approximately 1 year. So the web-based branch is feasible for older adults to complete independently on their own device, and it provides a valid reflection of in-clinic cognitive functioning. Very early amyloid-related memory declines can be revealed by assessing learning over days, suggesting that failures in memory consolidation may predate other conventional amnestic deficits observed in AD. And then finally, repeated digital memory assessments, increasingly feasible and uniquely able to assess memory consolidation over short time intervals, allow for early detection of subtle cognitive decline that's not otherwise observable using standard assessment approaches. There are a number of practical applications for these multi-day learning curves to clinical trials as well as research. And these applications may not just apply to our branch tests, but could be extrapolated to other sensitive digital measures and paradigms. First, they can be used in concert with biomarkers to develop individualized risk or prediction metrics and or to facilitate screening. They have the potential to be used to more rapidly assess the efficacy of novel agents in early phase trials. They could also be used to monitor cognitive safety remotely. And finally, they have the potential to serve as interim outcomes which predict later clinically meaningful endpoints. There's still quite a bit of work to be done, however, including validation in larger, more diverse cohorts. Additionally, we need to better understand how learning curves change over time. And finally, we need to assess the feasibility of this paradigm amongst those with some cognitive impairment. So general conclusions for today, I think digital remote neuropsychological assessments are no longer a future direction. Many are being developed, validated, and implemented into research as we speak. However, there's still much more to do to move digital neuropsychological assessments from a research setting to the clinical setting. I think in particular, should remote and unsupervised digital tools be incorporated into clinical practice, it'll be critical that a number of issues be resolved, including access to appropriate normative data from representative samples, more formal procedures to ensure data fidelity, and then consensus guidelines around how to provide feedback around digital testing performance and diagnosis. It's also critical that neuropsychologists are actively involved in the process of developing the standards for these tools and then developing these tools in and of themselves. And finally, it'll be critical to partner with big data scientists, as the breadth and volume of digital data will likely require advanced management and statistical techniques beyond what's required with our single time point in clinic cognitive testing. So with that, I'd like to thank you for your attention and acknowledge all the study participants and study teams from the Harvard Aging Brain and Related Studies, as well as my co-investigators in the branch group and our larger Harvard Aging Brain group at Mass General Brigham. I also like to thank my funders, and it was a pleasure being here today and speaking with you. Thank you.
Hi, I'm Shefali Singh. I'm an early career neuropsychologist. For accessibility purposes, I'm in my early 30s. I have dark hair. Today, I'll be talking about achieving accessibility through digital neuropsychology. I am a licensed clinical neuropsychologist and the director of digital cognitive research within the Bach Tech Lab at McLean Hospital and Harvard Medical School. And so today I'll be talking about a bit of background on why we do this work, and then some modalities for incorporating digital tools in neuropsychological assessment, sort of practically. Now, why do we do this work? Well, neuropsychological testing today is very sensitive, but not specific. It's very sensitive in that it can detect the presence of cognitive impairment and brain dysfunction. But we have a lot of problems establishing ecological validity. It's clear that we do our tests in a very controlled environment, and we're only really getting a snapshot of someone's cognitive functioning. Our tests also have limitations in that our normative data are limited. Some specific tests that we use only have an N of 15 as our entire normative sample, and they're typically on weird populations. Certainly in terms of accessibility and equity, there are differences in culture, even age. I know my Harvard undergrads would not be able to name this, this image right here. And in the absence of noise, our tests are actually quite noisy in that they don't account for contextual factors that are readily quantifiable with technology. So my work really aims to fill these gaps in traditional neuropsychological testing by leveraging digital technologies. And this will improve our ability to characterize psychological functioning in a way that is much more ecologically valid. So what are these digital tools? So I primarily focus on cognitive EMA, speech EMA, and context. EMA just means ecological momentary assessment. It is not new. It's been around for decades. But what it does is it enables us to get multiple time points of assessment to determine how someone is functioning outside of the clinic. And so cognitive EMA in particular allows us to measure cognition, cognitive fluctuations over time outside of the clinic. Speech EMA has been demonstrated to be somewhat of a proxy for cognition. And I'll talk a bit more about what I mean about that later. And then context. And so we have things like environmental or noise as context, and also psychological factors can be contextual as well. And then we have additional tools, which I don't use, but are being used in the space, like facial recognition software, things like computer mouse clicks, pressure pads, even texting speed. But these aren't all the tools. So here's sort of this high level image that sort of, I think, is a really, really nice depiction of all the things that you can use digitally. So on sort of the baseline level, we've got sensors, right? And our phones have most of these sensors. We can look at location, movement, phone screen, phone apps, a microphone. Low-level features, then we have our location type. Where is somebody? Activity type, movement intensity, phone usage, acoustic information. And then we have sort of what a neuropsychologist looks at, which are more high-level behavior markers, you know, psychomotor activity, fatigue, and these are sort of what we think of as symptoms that our patients endorse. And then we have a clinical state like depression or anxiety or ADHD, for example. Passive sensing is a technique that I think that is readily accessible using our digital tools that we have today. And what passive sensing is, and it was sort of like the bottom sort of level here. They measure physical signs using devices to sense properties of environments. They're great sensors, things like microphones, Wi-Fi, Bluetooth, keyboard sensing. And in addition, our Apple Watches, Fitbits, ambulatory blood pressure cuffs, all of these things are natural sensors that we often use every day. So my work really focuses on sort of the things outlined here, location, phone screen, apps, microphone, and then low-level features, higher-level features, and more clinical states. And so let's talk about cognition. So typically, you know, in a neuropsychological evaluation, we have sort of a long, thorough evaluation. But we're again not able to capture someone's functioning outside of the clinic. So being able to use digital tools that can be administered remotely make research in a lot of ways a lot more scalable and accessible and equitable. So I use Test My Brain, which my postdoc mentor Laura Germain developed, and it has millions of people in the normative sample. Again, not perfectly sensitive to specific cognitive disorders. We're working on that, but it is an excellent, valid, and robust and reliable tool that we can look at to see cognitive fluctuations over time. And as you see, the domains that we test are actually very synchronous with what we are measuring in our neuropsych evaluations. And how they work are, you know, we can administer these ecological momentary assessments multiple times a day. So here we've got, you know, 6 different time points. They can be fixed, they can be interval, they could be, you know, random. In one of my studies, I do it 6 times a month because the EMAs are a bit longer. So you can do lots of different things. And what you can get, and this might look complex, but what you can get is what we call very intra-individual variability and inter-individual variability. And just what that means is that each of these cells you can think of as one specific person. And if you're looking at something like daily negative affect, you could see that daily negative affect kind of varies between people. Like this person doesn't have a ton of variability, whereas this person might have a bit more. But then variability within a person, right? So it's that intra variability. And so we can look at not just how a person compares to other people, but also how they compare to themselves. And so that's really the sort of strength of doing EMA. It's like getting a longitudinal measure on yourself. So we used EMA in clinical and community samples and did a reliability study. And we found that after 10 EMAs, you get really, really robust within and between person reliability in both clinical and community samples and across different cognitive domains. So we've got visuospatial abilities, processing speed, attention, reaction time. So we've demonstrated actually that this is a pretty robust tool. Now, I'd like to turn to, so we talked about cognitive EMA, and now I'd like to talk about speech EMA. So what got me really interested in speech, and this is the group that I'm working with at IBM as a collaborator, is that speech, diagnostically, speech is sort of an objective measure of cognition. And I think it's one of, these next sort of frontiers that we can embark on. And so I've been incorporating it in quite a bit of my studies and have collaborations with both MIT and IBM, where we're trying to figure out ways that we can leverage digital tools and capture speech characteristics that might be prognostic indicators of disease. So, for example, in the AI program that this group that I'm working with predicted with 75% accuracy who might go on to develop Alzheimer's disease, which is actually a lot more accurate than a lot of the traditional measures that we use today. So this is just an example of what a speech EMA looks like. And so someone would just click it and they would say, the quick brown fox jumps over lazy dog, and we can look at a lot of different characteristics within this. So ongoing projects. So the first one is this Test My Brain digital neuropsychology toolkit. And we allowed the clinician to kind of, and again, this is all remote. It was sort of in the wake of the pandemic, our lab developed this where we allowed clinicians to choose their own battery from tests that they're actually used to using already, but these are just the Test My Brain versions. With really, really vast normative samples, and they can click them, create a unique battery, present it to a patient, and then get normative data. And we published a paper last year on this, and we still continue. I mean, of course, there's been a huge drop off since 2021, where people are now in person more, but we still have sort of over 14,000 test and survey completions even this year, and our factor correlations demonstrate that the tests are loading on domains that we expect them to, like processing speed and working memory and general ability. So this got me thinking that, yes, we can use digital tools in neuropsychological assessment. And how do we do that practically? So Laura Jermine and I came up with this hybrid neuropsychology approach, and it allows us to sort of demonstrate the three things here, which are equivalence, diagnostic clarity, and performance validity. And basically, this is a method that we can use to incorporate digital tools in neuropsych assessment. So equivalence, we hope to cross-validate and are working on cross-validating traditional tests with Test My Brain tests, so we can adopt more digital tools more frequently. Establish greater diagnostic clarity. So we'd like to look for patterns captured through digital responses that translates to specific cognitive impairments. So sensitive and specific tests. In addition, something we really pay a lot of attention to as neuropsychologists is how valid performances are. And so that is demonstrated on engagement. Now we have performance validity measures that can look at someone or they're sort of standalone. And there's also some that are embedded, and performance validity measures just simply state that someone is engaged in the testing that you're doing. Now, what we don't have a good idea of, and we do have some embedded measures within our tests, but we don't really know sort of intra-test variability of engagement, which could be an indicator of things like ADHD or even depression and anxiety. And so these performance validity measures, we can sort of use digital tools to make our performance validity measures much, much better and much more nuanced. And this will help us sort of determine cutoffs for identifying inconsistent response patterns, and we'll also be able to cross-validate our performance validity measures with standard ones to help identify poor or effort or engagement. Right now, we've got 31 participants total, and we're collecting this data in our neuropsych department. My next goal is really to develop rapid and scalable batteries. Scalable means equitable and accessible. in that you don't have to come to a doctor's office. And what we are doing is gathering cognitive, sleep, and activities of daily living data from patients in our short-term inpatient unit. And we're trying to determine the most streamlined battery that could be used for all McLean inpatient units. And the goal would be, instead of coming in for this full traditional neuropsych eval, we can kind of come up with things that demonstrate the highest signal that would make someone go towards needing a full neuropsych evaluation. And this will help us sort of understand the ways where cognition and mood and patient-specific data might relate to not only diagnosis, but also specific patient outcomes. Another thing that we're doing is, in developing these rapid and scalable batteries, we're now trying to do it sort of on a national scale. So one of my NIH-funded grants in rare diseases, we're looking at patients with metabolic disease, specifically phenylketonuria. Why PKU? Because those patients tend to actually have fluctuations in cognition, particularly verbal fluency and processing speed. And we can have some, and phenylalanine or PKU is really interesting because we can actually also get blood values like phenylalanine. And so what we're doing is we're doing cognitive EMA, speech EMA, mood questionnaires, and then while the day they're doing that, we're also getting blood phenylalanine and tyrosine levels, and we're doing 6 measures over 1 month, and they vary based on time of day. So we know morning, evening, or night versus day of the week. And the idea is to develop a cognitive and speech battery that's specific for patients with PKU. And that's what we've done in year 1. And then we're going on to validate cognitive and speech batteries and streamline them for rapid distribution. And the idea is that we can use this battery to monitor PKU patients over time in a more accessible way and develop diagnostic tools that can help determine prognosis in a way that's much more individualized and within person than what we can currently. The eventual goal is to integrate this battery into clinical practice and even clinical trials, and then disseminate this battery nationally, internationally. And this will really help in promoting equity and accessibility, especially for patients with PKU in other countries. Now, what we found so far, and this is just preliminary data, is that we have found that speech acquisition can serve as a proxy for cognition and does actually correlate with blood-based biomarkers like tyrosine. And so we can give tests that we give normally, like semantic fluency, and look at response times between words and frequency of words. And that was actually directly related to blood levels of tyrosine and phenylalanine. And this moves us towards a more precision medicine model where we can integrate our tests with both biological and biometric markers. I'm also doing using these tools in a big Bridge to AI grant through the NIH, where we're using AI and voice to better characterize and treat mental illness. And we're specifically incorporating speech and cognition at McLean and recruiting US-based neuropsych clinics, specifically those that upload in raw data in electronic medical records like us. And we're trying to see if we can find any discrepancies between self-report and clinical diagnoses. And so we're in the process of launching a pilot at McLean to see how voice and traditional neuropsych test performance plus electronic medical records can help characterize disease profiles. We are also launching an assisted living project where we hope to be able to provide traditional neuropsych evaluations, cognitive EMAs, and speech EMAs to better characterize and stratify degrees of cognitive impairment. So much of my work thus far that I've talked about is, of course, centered around improving equity and access, but then also it's really trying to use methods that allow us to develop specific, distinct phenotypes of patients such that we can better characterize, predict, and stratify severity of disease outside of our traditional methods of doing it. And we hope that what we find yields some more precise and accurate data relative to what we have so far. And then we're Also developing conversational AIs that can be used to chat with elderly individuals living in these programs. This can also just be used to improve mood and predict prognosis of patients who have mild neurocognitive disorder. And then I am working with IBM on methods for quickly assessing orientation using speech and asking even daily questions such as sort of a book club where they can sort of read chapters every week and kind of summarize them. So in general, I think it's important that in neuropsychology, we move towards incorporating novel digital tools that can improve equity and access. And we have long wait times, for example, in the clinic. What if we could provide Fitbits and Apple Watches and actigraphy monitors to patients during their long wait times for a neuropsych eval? That'll allow us to gather biometric data that could be translated using AI to text, and that could be integrated in a neuropsych report. We're also thinking about the ways that speech can be leveraged. So having patients blog, for example, prior to a neuropsych eval, so we can capture fluctuations in speech and relate that to performance on neuropsych evals. This would also be a great way of gathering sort of our history and presenting problem outside of our traditional clinical interview. Another thing I'm really excited about is potentially even developing a clinician dashboard and figuring out how we can incorporate biometric voice and digital cognitive data that can be readily used by clinicians. And finally, I want to be able to feed data summary sheets from a wide variety of clinics in a machine learning AI model. And we have so much raw data on patients and also, of course, their medical and psychological history, and figuring out how we might be able to streamline diagnosis and also our report writing. So those are some of the projects that I'm working on. And I think that by leveraging all of these digital tools, we can really improve equity and access, and in general, move towards a more precision medicine care model. So thank you so much for listening to my talk, and I look forward to the Q&A session.
Fantastic. Thank you both very much, Kate and Shefali. So I think we're going to have Kate and Shefali join us. We also have Dr. Marty Slawinski, who's going to be joining us for a discussion. There's Shefali, and Kate's coming on board. There we go. Fantastic. Dr. Slawinski, did you want to, they're going to pull you into the main screen. Did you want to introduce yourself real quickly? And if you have any comments on either of the talks, I know you were active in the chat earlier, so go for it.
Hi, yeah, I'm Marty Slawinski. I'm a professor at Penn State, and I do work in the area that Kate and Shefali were talking about. And just very quickly, I had two observations for each one of the presentations. And I was so glad I was here because what we're observing is, you know, a paradigm shift in how we think about measuring cognition. And I think the way in which the paradigm is changing was really nicely exemplified by both Kate and Shefali. With Kate's discussion, there are 2 points that stuck out to me that I want to highlight. One is her way in which she used construct validation as a touchstone to facilitate interpretation of the novel measures, but not really sort of using the in-person measures as a gold standard and saying for the remote assessment to work that it needs to be strongly correlated. So it's basically criterion validity is the target, showing the learning curves were related to the biomarkers, and the correlation with the in-person measures, that kind of tells us we're measuring cognition. But what I really liked about what Kate presented is that, especially with the data on the learning curve, it's really leaning into the technology. So too often, and kind of with COVID, we were prompted to ask, you know, how can we do what we do in the clinic with a smartphone or remotely on a computer? And that's fine. But what I really liked about Kate's approach is that she asked the question, given the technology, how can we optimize measuring relevant features of cognitive function, maybe in ways that can't be done by conventional measurements that are done in person? So I think that kind of prompts the question of what else can we do to understand cognition by using the technology of today and tomorrow. Piggybacking on that just very briefly with Shefali's presentation, I really liked the fact that she really nicely encapsulated the ability of us now to ask questions that we could never ask before by relying on conventional in-person measurements. Things like being able to evaluate cognitive fluctuations and examining short-term variability and change. And I think that this really provides an opportunity for us to ask research questions we've never asked before, like thinking about how to link putative risk factors to cognitive function in real time and across different time scales. For example, examining dynamic and time-ordered relationships between, say, social activity and isolation and cognitive function as that unfolds over the course of days, weeks, or months. So I'll just leave it at there. Those were there was a lot to like in both the talks, but those were what I wanted to draw attention to.
Thanks, Marty. Kate and Shefali, any comments?
I definitely appreciate the praise for the talks. I think it's interesting to see these different approaches. I really do liked how Shefali, with the Test My Brain, the ability to capture such large amounts of data, which I think is something that I didn't emphasize in my presentation, but to get people in the order of thousands, to be able to really get normative data as well beyond what we can achieve with our standard measures. There's sort of multiple benefits to this type of digital testing, but I do think that there's a lot that still needs to be realized to move these measures from the research setting where we can explore these interesting constructs and use this technology to really applying it in meaningful ways, for the benefit of patients. So I think that's sort of the next bridge of where we need to go.
Shefali?
Yeah, I agree. Thanks, Marty, again, and nice to see you also. Yeah, and Catherine, I really enjoyed your talk as well. And I think it's spot on to say that the real challenge is translating these tools. We see this in sort of companies all the time that are sort of getting a ton of different data and trying to figure out how they differentially detect signal. And so a big challenge is figuring out how do we integrate all of this data in a way that's also accessible to clinicians, because as clinicians, we're not data scientists, right? We have a very, very limited time with our patients, and we have to figure out sort of not find a needle in a haystack as I just heard it, but find many needles in a haystack and figure out which needles sort of yield the most signal. And so that's sort of the challenge, right? Is figuring out what yields signal that's actually valid and can be incorporated readily in clinical assessment. So
it's interesting that you're all making the comment about the value to the clinician. I mean, eventually, we sort of made this point yesterday that, just like drugs have a translation problem, there might be a little bit of a translation problem here too, from the research setting into the clinical setting. So what do you think we can do? So Monroe Collin made the point that, who's our keynote yesterday, one challenge is how we're going to eventually move these assessments into clinical practice, how to get insurance to cover these new procedures for clinical end users. This is going to be the key. So what do you think, excuse me, are the steps between where you are now? What steps are you going to need to take, and what barriers are there to get this into the clinic? I mean, the insurance company issue is always kind of squishy, so I don't want to get into that too much. But what do you think you need, just from a scientific perspective, to get from where you are now to a clinical end user who just wants to make a diagnosis?
Yeah.
So Shefali, you want to go first?
Yeah, I think that's a great question. That is one of the main reasons I joined the CPT advisory board at the APA, to try to figure out how we can get reimbursement for telepsych and teleneuropsych services. We have to demonstrate that our tests are actually, the digital tools are adding something to our evaluations. Right now, we have digital tools, we have digital technologies, but they're not necessarily sensitive and specific to diagnoses in the way our tried and true neuropsych measures are. So we have to prove that these tests are measuring constructs in a way that's better and more sensitive and specific to sort of characterizing and stratifying mental illness, right, or neurodegenerative diseases better than our traditional measures. And by doing that, it'll require large sample sizes, which is more feasible with digital technologies that are scalable and accessible. And then also the sort of validation against our tried and true neuropsychological measures that we use every day in the clinic. And then combining that with, AI and ML, where we can like look at electronic medical records and synthesize a lot of data, being able to use data in that way that we're able to better understand specific degrees and characterizations of neurodegenerative disorders and cognitive impairment, that's going to provide, I think, the bolstering that we need to insurance companies to show that these tests are actually better than what we're using now and thus should be reimbursed.
So before we move on to Kate and Marty for this one, let me ask you something. You just said large sample sizes, and I know that this comes up a lot, but you also mentioned your work with PKU. And so how do we handle this in the rare disease scenario?
Yeah. So the issue with the rare disease scenario is inherently they're rare, right? And so our sample sizes aren't that large, but Marty's work shows this quite a bit, and our work as well, where we find that is sort of the strength of EMA design, ecological momentary assessment design, in that it's sort of a longitudinal data set that you're getting. So on each person, maybe you're getting 20 data points, right? And so instead of having 3 people, you have 60 data points that you're analyzing, right? So EMA makes things much more sort of amenable to smaller sample sizes where we can figure out and streamline the digital batteries that we're making, and then use that as sort of a ground truth for developing larger and more scalable batteries that can be sort of more widely accessible. And the nice thing again about digital technologies is that they're iterative, right? So we can keep sort of making these batteries more sensitive and specific to disease as they progress.
Great, thank you. Marty, did you want to jump on that too?
Yeah, not being a clinician, I feel like I almost maybe shouldn't say anything, but 2 thoughts kind of piggybacking on some of the points Shefali and Kate made. The digital measures, again, not necessarily thinking of them as replacing what's done in the clinic, but what they can add. And if you think of two concepts, one is multi-stage screening in a public health context, that all we need to do is provide some predictive value by the digital measures, especially if they're low burden and very cost effective, to refer with some degree of confidence people who may need more specific and sensitive follow-up. So that's something we could think about trying to carve out a piece in the existing system for these tools, and thinking about a criteria, the number needed to screen to be able to identify a case as sort of maybe one criteria that could be added to the validation checklist that Kate provided.
Kate, your thoughts?
Yeah, no, I think that's a really good point. That I think that that the goal is not to replace the traditional neuropsychological assessment with these digital measures, but really like the idea of triaging our patient populations. And I think this is probably more applicable to more common diseases, like in the state the state of Alzheimer's disease, particularly in preclinical phases of different diseases where subtle or symptoms might be subtle or less demonstrative. And we can't get an accurate signal when someone does an MMSE in the office, at the primary care doctor, but we want to have some indication of whether this person, needs to be elevated to the next level of referral. And so I think I definitely see scenarios there where we use these types of tools to triage our patients. That the role then of how neuropsychologists and reimbursement would play into that, I think, is more complicated, because it seems like it would be more at a practice level versus at an individual level. But I think that's probably the scenario where we're really taking advantage of, the scalability of the technology by using it to create that funnel of minimizing wait times, getting the right people into the right departments in the hospital.
Great, thank you. One question, this is for everybody, is it just occurred to me as I was listening to these talks, but it applies obviously to pretty much all the talks we've heard. How do you guys all handle missing data? Because the more, that you're collecting a lot of data, so some's gonna be missing. So Shefali,
Right. So missing data is complex. Everyone knows that. And it's especially complex with EMA data, but also less complex in some ways, right? So a couple of things in the chat, we were talking about like what increases compliance, right? And it's hard, right? Especially when you're asking people in the middle of their day, you know, yes, it's greater ecological validity, but you're asking people in the middle of the day to respond to a mood questionnaire or a cognitive questionnaire, right? And that's challenging. So one thing that we found that helps, and this was in the chat as well, is like Marty said, compensation, but then also really sort of routine follow-up. And we actually have had we had 100% actually adherence in our PKU study because we had a lot of follow-up. My research assistant every day was kind of communicating with patients and participants and seeing sort of if they were able to complete the task. And so there's sort of this sort of psychoeducation that happens in the beginning of the testing or in the beginning of the study that says, you know, this is what sort of the demands are going to be, and they're sort of prepared for that. And compensation, of course, certainly helps quite a bit. With missing data, it's challenging, especially with EMA, because each EMA might be at a different time of day, a different time of week. That's how we purposely do it, to try to get that fluctuation. So what you can do is add on additional EMAs, or you can simply, but which isn't ideal because it sort of, you know, changes the sensitivity. But then what you can also do is sort of imputation, and you could try to just look at the person with less data. And that's not the most satisfying approach, but like, you know, that's sort of the beauty of EMA, is that if you miss one data point, you're still able to kind of get a pretty good picture sort of longitudinally. And it's not the perfect answer, it's not the perfect approach, but we often use imputation.
So let me ask you a question that would, so I'd like to hear both from Kate and Marty about how they handle missing data, but let me ask you this question first, Polly. What role does a statistician play in planning your studies, helping you to analyze the studies and that kind of thing? Where does that statistics approach come in?
So our collaboration right now on the PKU study is with IBM, and then in general, we have some incredible people in our lab that are excellent at statistics, and we're regularly sort of and machine learning, and so we're regularly consulting with those people as we're sort of planning studies. And so I think that that's just sort of a strength of the group, and being able to collaborate with IBM has been extremely helpful in understanding sort of how we can think about the data, especially with speech data. It's really complex, and truthfully, I didn't know much about it before starting this because there's a lot of sort of, you know, variables that we don't interact with regularly in neuropsychology or any cognitive research. And so that has been quite complex to navigate that. But in general, I think having close communication at the outset of planning these studies is absolutely right, that you need that, because it'll help you prepare for those edge cases where you're not getting the data that you necessarily want to capture.
Great. Kate, your thoughts on missing data?
Yeah, I mean, I think, you know, just like Shefali said, that missing data in the context of getting multiple repeated assessments is much less of a challenge than, you know, when our single time point assessments in the clinic. We have partnered very early with a statistician and have with her guidance an interesting approach to capturing these learning curves, where we actually look at the area under the learning curve, and that's pretty robust against missing data. We, interestingly, I think, have had maybe less challenges with adherence than I've heard from other people who do repeated testing. I think that might be in part as it relates to the population, and that, you know, we're mostly working with people who are worried well and have an vested interest in their own understanding their own cognition, and so that they have these intrinsic motivating factors of why they're participating in the research that are really not necessarily monetarily based. And also, I think that the design of the learning curve, and that people are getting the same material each day, I think there's a sense of mastery that people start to achieve. And so it's not that they're getting sort of a new task with each assessment that you know, is like a new challenge, but there is an element of it that they see that they're getting better and that they're improving. And so I do think that is a motivating factor, in that there's like an increase in familiarity, and then feeling like, you know, they've mastered the task by the end. We are working on one thing that we've gotten feedback from our participants about is that, you know, and so this is sort of the idea of not just adherence over the course of one burst, but adherence over a longer term to be able to follow people longitudinally, is that folks do want to know about, get feedback about their own cognitive functioning. One of the also the nice things about the learning curve is that, like, something like 99% of people get better. So we're at the moment, we're able to give everybody essentially, we're experimenting with giving everybody feedback, and that's largely positive feedback, and looking at whether that improves their adherence to, doing it 6 months or a year later. So I think there's a lot of different strategies to sort of at the outset try to minimize missing data. I think the communication part is key. We let people take the tests at the time of day of their choosing. We just ask them to keep it consistent, and I think that helps. And also, that's not necessarily something that can, if you're doing, other types where you want to get, different time points across the day, that might not be as feasible a strategy. But I think also allowing them to have some control over how they get it, so they can make the choice of whether they want to get it through text message or email, the reminders. I think also sort of adds agency. We have found, just to the question of, I think Tracy King is asking about patterns of compliance, that, and I think this relates to missingness, is that we do see that individuals who have some impairment, so like in the early stages of mild cognitive impairment, are oftentimes take longer to complete the entirety of our 7 days. So they might skip a day and then come to the next. So we do see that, that cognitive impairment is associated with reduced adherence. And that could be because they're struggling with the tests. It could be that they have ignored the reminders. They're not as tuned into their phone or their emails. There could be a host of reasons for that. But that's definitely a consideration that we've had about how we could potentially mitigate some of those effects and put in more structure or encouragement to get those people who have subtle impairment to be able to adhere to the testing schedule, as well as those who are normal cognitively.
Great, thanks. Marty, I think looking at the clock, you're going to finish this out. And there's also a question that you might want to address from one of our prior panelists, Liz Pena, about would planned missingness or computerized adaptive testing work here. So I'll let you get the last word in.
Okay, I'll be brief. I think what Tracy asked about and what we've heard here is that the missing data in terms of amount doesn't matter much, because we get so much data that the estimates are reliable. The concern is bias. And why are people not doing an assessment at a particular time? To give a little anecdote, in our studies, which are very demanding in diverse samples of older adults with and without MCI, we see relatively close adherence to completing assessments where they are signaled to complete the assessment. So the phone gives them a message. People with and without MCI complete those at about the same rate. We ask them to do an evening assessment, but we do not signal them, because we don't want to wake them up after they go to bed. They have to remember to do it right when they go to bed. Adherence for people with MCI drops off substantially, and that is now an unexpected measure of what type of cognitive function, prospective memory, right? So I think we should look at bias, but then also see, is there bias means there is information. So to see whether that information is predictive. As regards planned missingness in computerized adaptive testing, yes, this can work. I think it I would do it to try and improve measurement precision, but not necessarily to reduce burden, because I don't think people are not completing because there's one too many items or questions that we're asking in a particular assessment. But it's also very tough to do this in high frequency types of designs. But certainly that's a great point, and I bet in the future people will work this out.
Fantastic. Well, thanks to all of you. It was a really, really great discussion, as expected. Really thanks to the three of you, and thanks to everyone who asked questions and provided input. and again, you know, there's a lot of interesting conversations going on within the chat, and that's wonderful. You know, everyone's trying to help each other out here. That's fantastic. So I think thanks to our speakers and panelists, and we are almost on the home stretch, not quite. So if we can, we're ready for another set of breakouts, and I am sure these are going to be just as dynamic as the breakouts were yesterday. And so if we can have the slide, please, for introducing the breakouts. There's going to be a hard stop at 2:15. So today we don't have the luxury of rolling over to the following day. So there's going to be a hard stop at 2:15, and the discussants and their NIH partners actually really need those 15 minutes today to put the summary slides together. So so you're going to automatically be launched into your discussion rooms as yesterday. We tried to, you know, we weren't able to accommodate everyone, everyone's first request, but I think we did pretty well. If you are not automatically moved to a room, put a note in the chat which room you'd like to be placed in by topic, and then they'll, based on availability, try to shuffle you guys around. So the 4 topics are remote assessment in natural history studies, and then utility of remote research tools for clinical assessments, remote assessments in clinical trials, and apps and computerized measures. So those are the 4 topics. Our discussants are going to be the ones who are presenting. They're going to be sharing their screens like we did this morning and presenting their slides in our discussion that is going to happen at 2:30. So if you're in a room, if you're in a breakout room, you're going to be done at 2:15, and then you get a 15-minute break until 2:30. If you are the discussant or the NIH partner, you get 15 minutes worth of extra work. Thank you. And so we'll see you all in your discussion rooms, your breakout rooms, and then we'll see everybody back here at 2:30 to do the discussion of the breakouts, which Christy's going to be leading, and then some conclusions and synthesis that Christy's going to offer, reflecting back on the last 2 days.
Right.
So
welcome back. I think we have participants back, and what we're going to do at this point is go through and do a summary of our discussions in the breakout rooms, and we will start with breakout 1, which is looking for my breakouts.
I think that's me.
That's with Kevin. Yeah. Remote assessment in natural history studies. Kevin Kroll, Dr. Kroll is at St. Jude and has extensive experience in conducting natural history studies, and his NIH partner was Jackie Wright from NHLBI.
So we had a nice discussion and dealing with natural history studies, and just as a intro, you know, these natural history studies, there's quite a bit of variability in the, I think the types of studies, but the things they have common are they're often longitudinal and capturing growth and development and change in response to disease over time, and often over long periods of time. Because of the natural history of the disease, to understand the change in cognitive function over time, you often have to capture a lot of information to look at the environmental changes, the health-related changes, and behavioral changes, social changes, all of those things that may impact function over many years. And so unlike a clinical trial, which may span 1 to up to 5 years, some of these natural history studies may span 20, 30 years. The studies I'm engaged in are quite large, looking at either the childhood cancer survivor study, for example, which has over 25,000 survivors and probably 8,000 controls, and compared to other studies, which may be rare disease studies, which are lucky if they can get to maybe 100 survivors. But the point being that in order to study natural change over time, the data that is collected has to be collected consistently as much as possible, but over longer periods of time. And we talked yesterday about multi-site studies. Some of these natural history studies, because we're looking at change over time and in these populations, are kind of like multi-site studies that may range from 100 sites to, in my case, 20,000 sites, because we're following these patients in their natural environment. So it magnifies some of the issues we've talked about in terms of variability in environment, variability across patients, variability from a variety of factors. And when we factor on top of that the longitudinal nature of these natural histories, it becomes even more difficult, because you think about, collecting remote data over 10 years time, even if you're lucky enough to stick with the same measurement program, if it's a remote assessment, for example, computerized assessment, the change in technology, the change in the environment that may take place at the site or in the person's home can influence that assessment. And so collecting that information is important, and knowing the circumstances in which that information, that longitudinal information collected, is important as well. There's always this tension about using old and new instruments, and sometimes all of us in natural history studies have to update our assessments over periods of time as new instruments become available. Sometimes that's a little bit more difficult when the change in the instrument may not adapt the change in the individual's technology in their home. And so being able to provide that device control is a luxury sometimes in these studies. We talk a lot about cognitive domains for assessment, and some of these are similar to some of the issues that we discussed earlier. One of the points that came up, which is not necessarily tied to natural history, is basal level of functioning that is often difficult to capture in individuals with more severe impairment. So I think from our study and from our discussion, one of the great ideas that came up, if the results of this workshop could include some kind of portfolio of ideas and resources that could facilitate research design. Some of us with these large cohorts of cancer survivors have been through development of procedures that seem to be working in these natural history courses. Other individuals with rare diseases, are trying to get to that place, but they're starting with their local rare diseases and would like to get to a national level of data collection to increase the power and better characterize the variability that occurs in the natural environment, natural course of the disease over many years, and learning from those of us with more relatively common diseases would be helpful. Whether that's from device control and monitoring in hundreds or even thousands of environments, whether that's data collection through multiple sources where we need to capture not just the cognitive data, but the biological data that helps us to understand the change in course of the disease and its impact on cognition, or whether that's related to IRB and regulatory issues that are now a little more difficult when we're dealing with so many sites, the sites being the home environment sometimes. So those are, having some portfolio or some common reference site to go to educate each other on these issues, I think would be very nice one of the positive outcomes of this study or this workshop. The questions. Christy, you're muted. I don't know if you there you go.
Sorry.
That's okay.
So I feel like there is so much to unpack here, and I know that we don't have enough time to do this, but what I would say, what do you think is in terms of what would be most important as a first step to provide for investigators who are trying to develop these methods or employ them for longitudinal data sets? What's the biggest need? What's the most important thing?
I don't know if this is the most important because, but one of the things that came up, and it seemed to be a common theme across our discussion, was some of the early regulatory work and how to get, IRB and other institutional organizations on board for collecting longitudinal data, HIPAA, that includes HIPAA information, in homes over many years. That tends to scare the people on the IRB, and so it took us a while to get through there.
Yeah, that's we did have a speaker who had a family emergency and couldn't come, who was going to talk on IRB issues, and because I know this is a area that a lot of people struggle with, especially when IRBs don't know how to handle the really complicated ethical considerations with this type of assessment, with the digital nature, with the in-home nature, and with the ideas of implicit consent that we were talking about. So so
I think one of the other points that I would raise would be just the realization that whether you're dealing with 100 cases of rare disease or 20,000 cancer survivors, a lot of that data is going to be collected in different environments over time, and that environment may not be the same over time as the individual grows and develops and goes to school or moves out or changes states or wherever, and sometimes those things can't be controlled. And so you have to focus more on collecting and knowing the information that is in the environment rather than trying to control the environment. And so it takes a little more time to capture the type of device that they're using, who's in the environment, what's the situation, level of fatigue at that time of day, and all of those factors that can influence cognitive performance in the natural environment. And so that takes a little more effort to capture all that information. You may not be as concerned about when doing a controlled study that you have a little more control over.
How do you think this also impacts data sharing, especially in the context of the new data sharing guidelines and regulations from NIH?
Yeah, I mean, we're still working through some of those new issues, and I actually have not seen a whole lot in terms of information. We obviously provide that data and share that, but I think it has to be considered along with that information. Some of that data isn't necessarily included in a lot of the data sharing sets. We may have some of the richness of the data that captures the environment or the biological factors, the physiological health indices, the self-rated information, as well as the cognitive performance. But that variability in this year, were they using an Apple 12 or were they using an iPhone 15? All of that stuff is not captured as often in elite. Well, it's captured, but not as part of the data sharing.
Did your group talk any about costs and trying to factor in costs for these longitudinal studies when you might not be sure of how there's going to be variability in either the need to update software, hardware, or other platforms over time?
We talked a little bit about cost. Natural history studies are very expensive, and that changes over time. It depends on, I think, to some extent, the size of the study. If you have a manageable cohort of 50 or 100 individuals, you can monitor that a little bit better, and the cost would be more. You can provide the cost of the devices and the software and help regulate that. In some of our studies with 25,000 survivors, obviously we can't do that, and have to that then influences the type of selection of the available measures that can operate on multiple systems, multiple devices. Cost it becomes a much more complex issue when dealing with I think these natural history studies.
Well, thanks. Obviously, a lot of information here. We'll try to address your group's ask for some guidance and some resources.
Mm-hmm.
Thanks, Kevin. Thanks, Jackie. We'll move on to our second group. This is with Dr. Winia and Dr. Richard Benson of NINDS. This is NIH partner, and they were meeting on remote assessment in clinical trials.
Thank you, Christy. We had a great discussion. I want to thank everybody who participated in the session for their engagement. And Richard from the RLA company had a fantastic summary. So we're going to go through the slides that he put together. Again, Richard, thank you very much. And Dr. Benson, please kindly add to the topics that I'll mention. So we go question by question. I think this will guide some of the topics that we discussed. So the first one was with regards to research priorities to optimize cognitive assessment. These were some of the topics that came up. Some of them, as you see, are factors to be considered. Some of them are topics that we may have some proposed solutions, and some are just really open for discussion. But one topic is the factor related to patient anxiety. I know there has been some discussions about this previously, but in some cognitive assessments, sometimes there can be anxiety related to the fact that the deficiencies in some of the cognitive domains can be highlighted, right, as somebody's undergoing an assessment. So we study aphasia, for example, and sometimes folks get worried because their deficits become more pronounced, or they become more aware of their deficits when they're being assessed. And you add the technology aspect on top of that, that can compound anxiety on one hand. But on the other hand, having an assessment at home is also associated with a higher level of comfort. So there's potential benefit for anxiety, and there's also potential impact, negative impact on anxiety. So it's something to be taken into account. Technology allows for clinical trials to reach more diverse populations. What that means is, on one hand, again, this is another topic for which there's pros and cons. There is less access to technology within some populations. This is probably becoming lessened as time goes by, that most people have better access to technology. But it's still the case that some populations may have lower access to internet, lower access to technology, lower access to the platform. Conversely, it also allows for individuals who live remotely, who otherwise wouldn't be able to have an assessment in person, to participate in trials. So these are topics to be considered, because for some specific trials or for some specific studies, this can be leveraged as a positive aspect of teletherapy, because again, it can allow for some remote populations to be assessed. There's a topic related to technology here, which I'll reach in just a minute with regards to access, but again, taking into account that there could be more or less access in some areas. One other important topic that came up was the fact that remote assessments in general are different. So a assessment done remotely, or a therapy or a treatment done remotely, can not necessarily be equated with a assessment or therapy done in person. So if in the context of a clinical trial, if the pilot trial or the early phase trial assessed one method, that cannot necessarily be extrapolated to the subsequent trials because it might not be exactly the same. So it's something to be mindful of, because the alignment of those is important, and potentially planning in advance so that every single aspect or from early phase to late stage uses the same aspect might be beneficial. Going back to technology access, as we were talking about with regards to access, that is a key topic, of course, because there are different levels of access. One aspect that came up is the fact that in the context of clinical trials, if there is variability in technology access, that can be a source of bias that can be readily apparent or not. And if that becomes unbalanced between arms in a clinical trial, that can lead to differences that are not necessarily related to the intervention, but to technology. So this is a key aspect, and I would say probably one of the most important ones in the context of clinical trial. One topic that came up was the fact that whenever possible, ensuring that there is homogeneity in technology is desirable. This may entail providing the technology to the participants in the clinical trial. There's a cost associated with that, naturally, but having the same device to be distributed or the internet access allows for control of that variable. It also allows for alleviating some of the anxiety that I mentioned before, because it's not a new technology that, or it's not a technological access that is uncertain, right? It can allow for the assessor to guide the use of the technology, so therefore reduces the anxiety on the participant. These are some other topics that came up in the first area, but I think we can move on quickly to the next ones. What I'll mention here are the ones that are highlighted in yellow. This is the question in which we talked a little bit more about IRB, which is the fact that how do we monitor compliance in remote cognitive assessments or having teletherapy in a clinical trial. Naturally, it's very important to make sure that the systems being used are compliant. What that entails is that the IRB of the approving institutions need to provide guidance with regards to the platform that is allowed, right, from a HIPAA point of view, and also particularly that the platforms are secure so that people don't hack into the platform during the assessment. That's a very important consideration. There are several platforms. I'm sure many of you have had experience with trying one or the other, but some are approved, some are HIPAA compliant, so it's very important to use those. And then finally, with regards to the IRB, there are specific ethical considerations related to the fact that many times remote assessments are also recorded. So it's key to make sure that obviously the participants are aware of that. It's part of the informed consent. There's a great benefit on having recordings because that allows for subsequent assessments, particularly with regards to fidelity, assessing effect size, etc. But there is the loss of confidentiality risk by storing information that contains PHI. So video obviously is PHI, voice is PHI. So there's an added complexity there, and that obviously is something to be taken into account clearly with IRB. Richard, if you kindly move on, thank you, to the next slide. Another topic that we discussed was in terms of effective tools is the comparative validation with the existing tools. As we mentioned before, remote assessments are not the same thing as in-person assessments. How different or how similar they are in the context of a clinical trial is very important to consider. And they might be a different thing altogether. If the effect size is noticeable and it's associated with quality of life, for example, in a remote assessment, that is, something to be taken into account. But validation with regards to the known assessments and with regards to outcomes is paramount, right? I think we can move on to the next one. Thank you, Richard. Hybrid trials is an idea that came up, and it's a very important point as well. It allows for some elements in a clinical trial to be done virtually. Of course, clinical trials usually entail a large number of variables being collected at multiple levels or at multiple time points. So if some can be done remotely, particularly in the context of trials that have in-person therapy, for example, but some assessments can be done remote, that can allow for more data capture, better access. But naturally, it's important to make sure that whatever it's hybrid for everybody the same way, right? Because otherwise, you can ensure it can cause some degree of bias. If something is measured remotely for some individuals and not remotely for others, it can be different. I think we have just the last slide here, which is the fact that, again, as we think or we consider tele forms of assessment or remote assessments, it's important to make sure that we don't immediately extrapolate from what's been done in person, because the sensitivity of those tests with regards to capturing effect sizes might be different. And if the outcome of a given trial is based on measuring the effect sizes, whether it's in the early stage or phase or a late stage, if the remote assessment is not sensitive to capture that effect size, naturally that can lead to erroneous conclusions that an intervention is not good enough, when in fact it could be. It's just that the measuring tool is not adequate. I think that was the last slide. So that captures the highlights. We have a few other topics that I didn't talk about. And again, I want to thank everybody for participating.
Thank you so much for that terrific summary. One thing that I was wondering, and this kind of gets back to things we were talking about yesterday, with the hybrid design, which for clinical trials, I think many of us would agree that, you know, local recruitment from teams that are familiar has a lot of advantages. Would your group have said that centralized administration of the remote measures is a good way to go? Is that what you were talking about in terms of the centralized feature would be the intervention and the outcome assessment?
Yeah, we did talk a little bit. I'm sorry, I got a little bit of echo. My apologies. So we did talk a bit about that. We didn't get into any conclusion as to whether it's a better or worse, but as being a factor to be considered. In the context of administering therapy, for example, or assessments, sometimes if they are they're different, it may allow for therapists, for example, that otherwise wouldn't have been available at different sites to administer therapy far away. So one example that came up was a trial that I'm involved with, which is associated with a particular form of speech therapy where not many therapists are familiar with. So having somebody who is in one state being able to administer that therapy for somebody who's far, far away is great. There is that advantage with regards to the centralization, but it does reduce the potential benefit of later on. If you do a clinical trial, you want to make sure everybody can do that, right? And then if you just do it in one side, you can't really assess, particularly in early phases, how well that can be adopted by folks that are not all in the same place. So we talked a little bit about that without any definitive, better or worse, but the pros and cons should be considered.
Lots of pros and cons there, I think. Let me think. I had another question. I'm trying to think. We also talked, obviously, just with the choir group about this issue of consent and IRB. And I think, it's a shame we lost our speaker on this today, because it is an area that when I was looking to see for some guidance, there are really lack of publications to guide people who are using remote assessments. And I know most of what's out there are highly individualized information from single institution IRBs. So were you thinking of a guide for investigators when they're going to IRB and they have to use a single IRB if they're in a multiple site study, about where they start or what they should be thinking of when they are trying to kind of come up with an informed consent that will capture these issues and also be relevant to people at multiple institutions that may have very different developing approaches to how to handle this?
That is a great suggestion. We did not talk about the establishment of a guide, though I think this would be quite beneficial in terms of even best practices, so to help the IRBs, right, in terms of understanding what the risks are associated with this. On a personal level, when I started a trial with teletherapy right at the beginning of the pandemic, it was an adventure, right? Because there was no experience from the IRB, and understandably, there were a lot of concerns about the platform, about the access to therapy, not leading to bias, not reducing the access from some populations to therapy. All I can say here is that I think key aspects certainly are the fact that one should use a platform that is HIPAA compliant and that is approved by an institution. I think a big issue is people hacking into the session. The second is the fact that recordings used in the platform are not stored in the cloud by the platform, that do not belong right to the investigator, that belong to somebody else. And then certainly the informed consent with regards to the fact that things are recorded. But in addition to those, I think having a guide would certainly be a public service, right? It could certainly help a lot of people. So I think that's a great suggestion.
Well, that will be for the investigators to develop on your own, but I think it would be a good service as people have discussed today. Thank you so much. Thank you to your group and to your discussion.
Thank you.
And now we will move on to group 3, which was Dr. Michelle Chen and her NIH partner, Dr. Jana Belzer-Ehrlich, and they were talking about the utility of assessments, remote assessments to clinical assessments. So how can we make these remote assessments clinically meaningful to our participants and patients?
All right, so in our group, we were discussing issues in selecting remotely administered cognitive measures for research purposes with clinical populations. And at first, the pre-prepared questions we have are really, thinking about, what is it important for, our tools to have established clinical utility for patients to be used in research. But then I think our group really kind of started talking about really the other side of it. We really talked about this is really a chicken or the egg question where, first of all, is it important for tools to already have clinical utility to be used in research? But then the question is also, if it hasn't been done in research, how can that be validated for clinical populations? And then we were discussing, ways to validate remote tools for that can be used in clinical practice, and what are things that would be needed, for it to actually be translated into clinical use, and also something that can be paid for, get reimbursed. So then one thing we talked about is including relevant stakeholders when considering clinical utility of remote tools, involving like policymakers and payers who will, be the ones that would deciding whether they can pay for for this clinical service if it's used clinically. That they also need to be part of the discussion for us to really be able to determine that. Another thing that we talked about is, if we're selecting tools for, different clinical populations, is it important for that tool to have already been validated in your particular population of interest? But then that makes for clinical populations that might that are, for the rare diseases, for example, that, haven't been talked about, sometimes it's hard to already, have something that already been used in your population. So then is it, how minute should the groupings be? And can we create larger groups of similar populations when we think about, whether, this test has clinical validation? And then also thinking about the feasibility of tools, right? If you're selecting tools where either based on like the user experience or design, or the demands that it has, thinking about, what are the clinical characteristics of your clinical population, and then selecting tools that are actually appropriate, and then also selecting tools that can actually get at the cognitive domains that you're interested in, especially in this remote setting where most of the tests are going to be brief. So then actually selecting the ones that are the most clinically relevant for the patient population is important. And then another thing that we talked about in our group is return of research results, and how that is now often an expectation of research participants and also in patients as well, where, with patients, like oftentimes they have really almost instantaneous access to their medical records and their patient portals. And the same thing can also be true with participants where they might be expecting a return of research results. In clinical tools, dashboards can be built in where the patient can, directly look at their performance or their results instantaneously. So then we talked about, what the pros and cons are. Pros are, for patients, to empower them to, have their own information, and then be able to, to use that to understand their disease. But then the con would be, if you don't have adequate resources for interpreting the results for them, either providing them with norms, which oftentimes a lot of these new remote tools don't really have adequate norms, then, how do you present this information in a way that is not going to be misused? But then, but is it also ethical to withhold that information just because you don't necessarily know, how to make the results most useful, when people might want all of the test results, even though some things might not be, some variables might be more significant than others. And then finally, we talked about, what are, how should remote assessment tools be validated? So one thing is validating against traditional gold standard measures, which is really important to establish construct and concurrent validity. But then we also talked about how that's not necessarily enough. I think those of us who use these remote assessment tools recognize that these tools can have a lot of advantages and in some ways can be better than our traditional tools, right? So just validating against this traditional tools that we think, has limitations is not really enough, and kind of looking into other things that can be more indicative of real world outcomes might be more adequate. And then we talked about how using passive sensing methods can be kind of integrated into these remote active assessments to understand the individual's context and fluctuations in their diseases and different things in their real world, which is really what these remote assessment tools are really great for.
Thank you for that summary. I'm really glad that your group brought up the return of research results, because I think that's something that we just have had a little bit of discussion of in previous talks or allusion to, but I think it's really important. And I'm wondering, if your group thought about what, where to start with that, especially for some of these measures that maybe collecting a lot of data, especially in the digital space, right, where there is lots of data being collected. It is potentially less expensive or less cumbersome to return results to patients, at least partially or in a digital format. But how to make meaning of that and to provide resources to patients, it would be another leap forward. Did you all talk about sort of where to begin with that? What the lowest measure about, or the lowest threshold of what research participants can expect when they have remote assessment should be? Or did you all sort of say, this is important, but we're not really sure how to do it?
I think we're mostly in the kind of latter camp of thinking about that. It's something that's important, but I think there's a really good consensus as to what's the best way to do it. But I think starting out with just the scores are kind of the most analogous to kind of our I think gold standard clinical assessment will probably be kind of the best start with, and then being able to eventually integrate algorithms into that can directly calculate different digital biomarkers that can be shown in a participant dashboard will probably be good. Will probably be a good next step. I know that for example, when you use wearables, there are certain platforms that that can calculate the digital biomarkers. Like if you're trying to measure sleep, and they can directly give you the sleep parameters. So can we do something similar with these measures that capture not just kind of the traditional summary scores that we use for paper and pencil neuropsych, but then also more about processes that are involved underlying those performances. I think that's kind of the opportunity of it, and then returning that is helping people understand that. I think that's kind of the next challenge, because you don't always have the resources to have an expert neuropsychologist to kind of give them feedback individually. So how to do that in a scalable manner is a challenge.
I like that idea, and to me that makes a lot of sense, and I think it is also something that is potentially really palatable to a research participant who may just want to know, like, if you say, hey, we noticed that in the morning your performance on that measure of attention is a lot stronger than it is in the afternoon, and that provides intra-individual knowledge that someone may be like, yeah, I've noticed that too, or maybe I should start to do my most important tasks in the morning, things like that. That's usable knowledge, even if you don't have normative data for every task that you do. But I'm sensing an opportunity here for you or others in your group to really talk about return of results in this space, in offering some information or guidance or thoughts to the larger research community on how to move forward with sharing research results.
No, I think that's a really good point. Something actually we also discussed is also that even without adequate norms, like being able to look at longitudinal changes, based on this individual's baseline, it can be really powerful.
And I just want to note that we've had the recurring theme about how to validate these measures and what are our appropriate standards for validation. So I appreciate that your group had a nuanced conversation about that validation, that I'm sensing, and I'll mention this when I talk about the summary of the workshop, that people are really thinking that with remote measures, validation needs to involve more than just reference to our traditional measures.
Yeah. So I'm sorry. What is the question?
In terms of more than the validation can't stop by just saying, like, this is how our remote measure is associated with something that purports to measure the same skill in the clinic.
Yes.
It sounds to me that people are saying that's not enough. That's not enough to just compare.
Because we were discussing that. That's really, that's an important first step, but it's definitely not the last step. And then the advantage of remote tools is really be able to kind of look at real world processes, which traditional clinic tools aren't really great at doing that. So then how can we connect to outcomes that are actually functionally meaningful in the real world, that is related to some kind of real world behavior change that's meaningful for them, and depending on the population, would tell us like more of, this is the added benefit of remote tools, and that it's not just about, doing things in a different format, but actually having added value.
A great way of putting it. Okay, thank you to group 3. And for the last 15 minutes, we're going to go on to group 4, which was led by Dr. Marty Slinsky, and talked about apps and computerized measures, along with me supporting. But I will take a back seat and let you talk about our summary, Dr. Slinsky.
Great, thank you. Can everyone see the slides? Okay. So this was a great session, and part of what made it great is we had a nice mix of people who have considerable experience doing remote cognitive assessment, as well as a nice mix of people who are wanting to get their feet wet in this space, and were able to share some of their concerns and thoughts. So there are a few themes that I'd like to highlight, and I'll do that relatively quickly. I think one of the most important points that was raised is just how vitally important accessibility is. And the example was given that an individual working with a certain clinical population are unable to provide response inputs in mobile devices in standard ways through finger tap and such. So we really need to think about and be mindful of designing assessments that are potentially adaptable to maximize accessibility and inclusion, both on sort of the response input side as well as the stimuli presentation side, the instruction side. This touches on themes you've heard throughout, I imagine, these 2 days on the importance of language and culture and how to think about all of that. Connected to this point was the recognition that there aren't necessarily measures that exist that are maximally accessible and inclusive. So there was kind of the uniform, or at least the vigorous endorsement of the ability to be able to adapt and build on existing measures for local use cases. And there's a recognition that we'll need to balance concerns about standardizing assessments with the idea of innovating to improve accessibility and inclusion. But I think, the sentiment was that one single provider can't possibly come up with sets of assessments that are going to be suitable out of the box for every particular use case in every particular environment. So giving people the opportunity to make those adaptations and innovations for the field is important. The next theme was sort of the importance to assess, and I'm calling them time-varying variables that can influence cognitive performance. You heard this piggybacks off of the discussion on the first breakout session, and it centers around identifying common data elements for remote assessments, and just something to pay attention to with properly designed software. It's often possible to capture some of this information passively. So I don't think we can ask people to sort of find the settings in their smartphone. I still don't know what kind of smartphone I have because I can't find it in the settings. But we can capture a lot of this information passively. Maybe in important ways, we can capture information about screen size. We can capture information about operating system versions. So anything we can capture passively, we should do that. I think it's also what came out was the importance of assessing proximal, in the moment, individual level variables that we need to be able to contextualize performance. So I shared with the group, I think I may have shared this with the broader group as well, sets of questions that we always ask individuals where they're located when they're doing the assessment. Is anyone else in the same room or around? And then afterwards, did anything interrupt them while they were taking the test? So not to say that these are the best questions, but something to help contextualize the performance in the moment, as well as this didn't come up too much, but it again relates to other topics, sort of broader contextual influences that might be slower acting, such as environment, like knowing where people live so you can assess environmental exposures, look at neighborhood features, and then track changes in residents. There was a fair amount of time talking about the need to maintain continuity for valid data collection in the context of a rapidly changing landscape, a technological landscape. There are new devices that are released all the time, software updates, technology updates, and single providers who might be providing the assessments you're using may make decisions to shift elements of their platform, which can affect your data collection, or update measures in ways that could affect your data collection. And I think as we think about how to build an ecosystem that the field can use in ways that promote both standardization, rigor, transparency, flexibility, we really need to think about maintaining open and transparent processes for developing and validating the software and the assessment tools, and making those resources available. So being able to inspect and see the underlying code for the assessments is important, as well as maybe decoupling the validated assessments from delivery platforms, so that a set of validated vetted measures could be delivered through different software platforms, whether it's REDCap, Qualtrics, or commercial EMA apps that could open a web view. And then finally, we spent some time thinking about normative data. There were some great questions asked. Where did the normative data come from? How many people are in the normative sample? There are some remote digital assessment platforms that have thousands or millions of people, but again, where do those data come from and how do we use that? This led to a discussion of, well, what do we use the norms for anyway? And perhaps rethinking what we want from normative data in a way that parallels the tone of the conversation today, of maybe rethinking what we want from remote cognitive assessments, not just to recapitulate what's always been done in the clinic, but maybe what can we do now with the technology and this mode of data collection that we couldn't do before. So it becomes more feasible when we think of cloud-based assessments and sort of automatic data depositing, to be able to develop norms that we actually might want to use, that might be sort of geographic or local norms, the ability maybe to norm and correct for technical variability across devices. And then the other piece is maybe thinking of really what we want from normative data. To my understanding, that all available norms are usable only once. You can only use them the first time a person takes the test. They don't apply to the second time or the follow-up time. And because when we develop norms, we typically just give the test to the normative sample once. So we don't know how to think about repeated testing and application of norms, but that could become possible with digital technology that allows us to do assessments more frequently, sending data to central repositories where we can rapidly and continually update normative data. Also, we have sort of norms from this century, being able to have time-relevant norms. So, because it is so hard to do this, once we collect a normative sample, by the time we do it, maybe things have changed such that these norms no longer apply, or they don't apply for very long. So instead of thinking of norms as something that resides in a manual printed on a page, norms are information we extract from a dynamic database, which is continually updated in ways that can provide relevant information for a variety of use cases. So those are the comments I had, and I'll stop here.
I was in this session, so I was party to a lot of these discussions, and but I want to get back to the point where this tricky point of the idea of how do we maintain continuity in these measures in the face of rapidly changing technology. And it used to be that if you employed one version of a traditional measure and you wanted to keep using it after an update, all you had to do was sort of stockpile the forms that were used. And potentially you could do that from getting them from other investigators or purchasing them all at once when the forms are about to change, but this becomes a lot more tricky in the remote assessment space, especially when you're using any kind of digital software or apps. And so I'm wondering, I have two things that came in my mind. Does this mean that you're thinking that researchers should have some flexibility to adapt or maintain a version of an app or a measure for use in their own study? For example, if they're doing a longitudinal study and they don't want to potentially change platforms, change software midstream, do you think that developers should really have this in mind and be prepared or thinking about how to decouple their platform with from the established platform and be able to make these available? And
Yes.
how does that go into, you know, intellectual property and ideas and copyright things about some of this technology?
Well, I mean, right. So if the goal is to develop proprietary tools that you can monetize and make money, then basically you should do that. And that would be the single provider model. And then great. You know, I think what this question comes from is the recognition that that is inherently risky, because if you do that, then you are sort of at the whims of whatever decisions that provider makes. If they want to do an update, or they want to change the font, or they want to make some sort of change, you are stuck in with that. There's also, they could change their business model and not do remote testing anymore, right? So there is some potential risk for that, and there is a way to decouple this through cloud-based assessments that can, right? It's easier said than done, but I think what we don't want to do is lock the validated assessments behind native apps that are proprietary, that are not open to the community, that investigators and clinicians have no control over. The issue about sort of maintaining standardization is very important, but there are potential solutions for building in checks for being able to do that.
Do you think that this will require creative solutions in terms of how to fund the development of these apps and platforms so that kind of flexibility is possible?
Yeah, I mean, I think that there are some models in sort of the tech space and the data science space. So R is kind of a model where there is a platform. You know, it's different than what we're talking about here. There are other examples we can kind of point to. I think establishing a robust community for that where we can cultivate expertise in being able to do this, kind of like in these kinds of workshops are a step in that direction.
Fantastic. Well, thank you so much for the summary, and I wanted to thank everybody involved in the breakouts for such a great discussion, and particularly to our moderators and NIH partners for providing such a quick turnaround and in terms of summarizing the great discussions that you had. Mindful that it is Friday afternoon and we are coming to the end of a lot of content, I am going to jump straight into the summary of the workshop and then hopefully let everybody go with their weekend a little bit early. So I'm going to share my screen unless there are any other questions, which certainly people should feel free to dump in the chat as I'm getting my screen going. All right, can people see that? I hope.
Yes, looks good.
Okay, great.
Yep.
So summarizing the workshop was a challenge. And so, you know, Adam told me that I had 4 slides to do this, but as a good neuropsychologist, I chose to interpret this with a 95% confidence interval. And so I have a few more slides and I will try to get through them as quickly as possible so that everybody can go. But if you have watched all or even most of the content over the last 2 days, the first statement on this slide here is self-evident. Remote assessment is here to stay and is allowing exciting innovation in a field that has used many of the same methodological approaches for many decades. It's an exciting time to be involved in research or to be observing research in this field. And I really agree with Dr. Smolinski's observation earlier today that we're witnessing a paradigm shift in the field. One theme that emerged many times is that remote measures should be considered new measures, even when they're adaptations of traditional paper and pencil measures. Dr. Cullum and others noted that many of these adaptations used in teleneuropsychology have very strong reliability, good validity and acceptability from the participants. And that this is particularly true for language-based and verbal reasoning or verbal memory tasks. There is additional work that is needed to either make better adaptations of visual spatial skills, tasks, and executive functioning measures. But there's also the idea that that some measures are not really lending themselves to just straight adaptation from our in-person, and that we really need innovation to develop new measures that will better capture these cognitive skills. In addition, we've heard from multiple participants that some people have to start from scratch. And we really have to start to scratch to know how to best measure some cognitive skills, as I said, but also in different populations and for different purposes. In listening to our speakers, I was struck by how these new methods may actually do a better job at answering some research questions than our traditional methods do. We focused less on analytic methods today, but the implication of these new methodologies, especially ones that provide more data points over time and just more data, period, is that we'll need to use innovative data analytic approaches in this remote assessment space. And then finally, I wanted to just say it's impossible to separate remote assessments from technology, even if all you're doing is translating an in-person assessment over Zoom. And this really opens up a lot of issues of access, of literacy, for technological literacy, as well as language literacy, and the need for frequent updates in terms of technology, as we were just talking about, how we manage to innovate and update and change these remote assessments with changes in technology. I was reflecting on all the ways that remote assessments can differ from our traditional evaluation methods and how difficult, frankly, it was to summarize these main points. What I concluded is that remote assessments involved changes in three important domains from traditional tasks. And first, the testing platforms for remote assessment often diverge from our traditional measures. And this is the domain where I think there's a lot of choice and variability. We heard about different tasks that involve different devices, accessories like pens, or that are web-based, that are wearables. So there are a lot of different testing platforms, and I think they're going to continue to have innovation in this space. The testing procedures themselves are often very different from our usual measures and provide a lot of opportunity to change the traditional paradigm. I was struck by what several participants highlighted in the talks and discussions about how customizable the procedures are to fit different research populations and different objectives. In contrast with our traditional measures, many remote assessment procedures can be highly customized and with high precision to control the length of exposure of stimuli, to measure response or reaction times with much greater precision, to capture multiple data points simultaneously that would be really difficult or impossible to accomplish with traditional methods. And finally, the testing environment is really the Wild West space, as it was described several times. And our participants noted that there still isn't good consensus on how much investigators should be recording about differences in the testing environments and how much that variability in the environment may contribute to differences in performance. And I just say differences in performance because I think the other thing that struck us is that they could be that we like to see the differences in performance as a function of this environmental context because it helps us shape and understand differences in individual performance over time and in patterns of performance. So I then thought about what opportunities we have for remote assessment and what some challenges are. I was really struck by what Dr. Singh and other speakers noted about the potential to better track intra-individual and intra-individual variability with remote assessment methods. When you're able to assess cognition in different contexts, perhaps those that highlight the participants' convenience, their comfort, their alertness. We have the potential to capture data that allow us to analyze patterns of functioning within and across individuals as they vary according to a number of external and internal factors. I think another key opportunity is that we have traditionally, especially as several speakers noted, had difficulties in access for cognitive health services. People often have clinically long wait times that can be difficult to involve this cognitive data collection in research. But monitoring cognitive health and screening for specific difficulties using remote methods is potentially easier, cheaper, and faster. We talked a lot about centralized models of remote cognitive assessment that may actually be really feasible in terms of less cost and being more efficient in multi-site research designs that would like to involve a cognitive testing component. In addition, we heard a lot about precision medicine opportunities with remote assessment and the opportunity to involve cognitive health metrics where traditionally they have been very difficult to involve or very limited in the information they were able to provide. So when it's quicker and easier to measure cognition, as we've heard, we are able to associate cognition with changing biomarkers that normally we wouldn't be able to capture real-time changes with. And this becomes not only possible, but becomes associated with real data that is meaningful to researchers and also to patients. And then I think another opportunity is to increase diversity and inclusivity in our research participation and sample. I think many people said that equitable inclusion is the goal, but this is still a work in progress, that we still have a lot of features to make sure are accessible, reach different populations, and that includes both geographically diverse populations, racial and ethnically diverse populations, and important speakers of languages other than English. But I think what I was excited about is, again, highlighted by a number of speakers, remote measures may actually provide us with better normative data than we've ever had. Not only up-to-date data, but data on much larger groups of people, much more diverse groups of people, and groups that have different differences that we may be able to describe and highlight for the first time. And then I think another important thing that we heard about was that there may actually be an improved user experience with remote measures. And increasingly, it's the opportunity to check in with users about their experience and then be able to refine these measures based on feedback that is really exciting about these measures. And then I just want to conclude with talking about some challenges and where we go from here. We really talked some about how we need to work towards convergence in clinical and research utility for at least some of our measures. And this is really for practical reasons. If remote measures aren't clinically useful or covered by insurance, they'll be harder to adopt into clinical trials at scale. Several speakers noted that while there will always be room for purely research measures, for remote assessment to gain greater traction and become more scalable, there's a need to establish clinical providers' familiarity and trust with these tools, and for third-party payers to reimburse for their use. So this is a space that we'll really need to explore in the future. I think challenges still include inclusion of some populations, as I noted on the previous slide, native speakers of languages other than English. We lack tools for them. We lack traditional tools as well as remote assessment tools, but this is a space that really has a lot of opportunity to include people who speak languages other than English. We also need to really think about including individuals who have low technical or language literacy. Again, you can't separate remote assessments from at least some technological literacy that is required. And we really need to make sure that even in this age of what we think of as ubiquitous or near universal access and near universal use of technology, that we have people that are going to be hard to reach who don't have familiarity or access. In addition, I think we have a lot of choices in this space. There's lots of innovation. There are many different tools, many different platforms. And I feel like we heard people say that we're ready to kind of curate some ready for use methods from among these many newly developed options. This really leads to the discussion of a need to develop and define common data elements across remote assessment measures so that ultimately we can develop a process to compare different results using remote assessment methods across platforms and across populations. I wanted to just list a couple of other challenges. We heard so much about how to achieve a balance between standardized and naturalistic methods. Standardization has really been our research standard, but in the remote assessment space, this can be expensive and it can create less inclusivity. BYOD, bring your own device, approaches are more likely to include individuals who are using the platforms or devices with which they're most comfortable. But this also creates noise in data that investigators don't yet know how best to capture or control for, or even what it means for performance variability. And then I was also struck by the idea of planning for and addressing the unexpected. So this can encompass safety and reporting events that are possible and that need to be prepared. I think that this is another area where there's opportunities to talk more and provide guidance to investigators to be able to follow research and clinical ethical procedures. And again, the idea of researchers being mindful and plan about what participants are implicitly consenting to when remote data is being collected, including not only recording of the data that you're collecting, but also what might be recording other people in the home, other activities in the home, and what this may mean for participants when they consent to participate in a research trial with remote data collection. And then again, we talked only a little bit about missing data in this space and how tracking reasons for missingness in data, particularly if it's non-randomly distributed, and ways to address missing data are probably really needed to be developed. And finally, I just wanted to give a nod to our many discussions about validation and standardization with remote assessments. Many of our participants noted that the measures we typically refer to as gold standards are sometimes really gold standards, and sometimes they're merely the measures that we have available and are used to using, rather than measures that have the best data supporting validity and the best normative samples. So a lot of people, and I really appreciated this idea of moving towards thinking about validation with gold standards to more corroboration with existing measures, and really to start with how our remote measures may corroborate with what is already known, and the recognition that may not be the most appropriate way to validate these newly developed measures. And so in that way, our approaches to validation, many speakers noted, may vary by cognitive skill or may vary by population. And in addition, standardization may need to account for a larger number of factors that are in that testing environment space and the testing paradigm space. And that would include context and devices, as well as consideration of groups of health disparities, or cultural or native language factors. And I think one of the big questions that came from this workshop were, do all of these little factors that we usually control when we use our traditional methods make a difference in performance? And if so, what are those factors? How do we account for them? And which factors are the most important for us to track, particularly in things like common data elements? I think along the lines of standardization, we heard a lot about sort of rethinking the approach, and this process be a dynamic one and revised in real time as our samples grow. And I think the main thing that we heard from speakers was that constantly renorming in the context of unique software updates and coming up with a completely new normative sample is probably feasible. I just wanted to also note that we talked a lot about rare diseases or small samples. And so what I took from those conversations was that just like with other areas of work involving small samples, reaching out of your silo to partner with another rare disease group may be particularly helpful in the remote assessment of cognition space to establish feasibility, reliability, and specificity of a newly developed measure. And then I think another idea that was mentioned by some of our participants was sort of stacking data collection with multiple measurement approaches, so that you get a lot of intra-individual data that may make up for a lack of a large sample. And then focusing on establishing metrics for reliable or meaningful change, not just in small samples, but in larger samples as well. So I wanted to just, in closing, thank the planning committee. This is a huge endeavor that we have been working on for the last 6 months. And so I appreciate the team approach that we use. And then I know I speak on their behalf to thank all of our participants, panelists and speakers who came and contributed their expertise and their time to share with everyone today. So I think we have a few minutes for questions, but if there are any, but otherwise I'm going to stop sharing my screen. And I wish everyone a fantastic weekend where you get an extra hour of sleep. I know that I will be enjoying that this weekend. Adam, did you want to say anything else?
No, it's been a really fantastic workshop. Thanks to everyone. Really, what can you say? It's been a really wonderful 2 days. Christy and I have a ton of work to do now, as does the planning committee. But, you know, this is, if it were easy, we wouldn't have to do a workshop. So hard problems and a lot of very, very thoughtful input. We've had some really wonderful comments. So thanks to Christy, the committee, all the speakers, breakout leaders, everybody. Their data blast, it was just one thing, you know, I felt like I was sort of getting hit from 20 different directions. So awesome.
Take care, everybody.
Have a great weekend. Thank you.
Bye.